Morning, everybody.
Morning.
Yeah, we got that. My name is Onjalé Price, or Anji. I'm the director of the Woods Hole Partnership Education Program, and I'm really excited to welcome you all to the 2024 symposium with the students that I've had the pleasure of hanging out with and getting to know this summer. A couple housekeeping things. I want to ask you all to take a moment to the audio in this room. You don't have to use a microphone, but everything in the room is the chatter, make sure that there's no cell phones that are ringing, and for restrooms, if anyone needs a restroom, they're out of this door directly ahead. People need the restrooms if necessary. If you do need to leave the room at any time and come back in, we ask that you use that door so that you are not disrupting the presenter here. Okay.
There was a request that you used.
Oh, there is a request that I use the mic.
Yeah, you can't hear up on the back.
Oh, okay, great. Okay, great. Thanks. There is coffee and some juice in the back at the break at around 11:15. We will have some more snacks, and then we have the lunch at the end. For those of you who are on Zoom, you are muted and your camera is off, and we would prefer that you stay that way. If you have any questions for any of the presenters, please put them in the chat. And when it's time for questions, Autumn will read any questions aloud from the chat. All right. So we are all here for the 2024 Woods Hole PEP Symposium. I'd like to give a little bit of background about PEP and how it started. So back in 2004, the Woods Hole Diversity Initiative was created, and that is the leaders of Woods Hole scientific institutions. They came together and recognized that they wanted to do something about making their community and the scientific institutions more inclusive, more welcoming, and more diverse. So they created the Woods Hole Diversity Initiative, and shortly after realized that they needed some other people to help do things, because the leaders of 6 institutions are very busy. And so they created the Woods Hole Diversity Advisory Committee. And the goal of the advisory committee was to provide the diversity initiative leaders with ideas, suggestions on how to make the community more welcoming, inclusive, and more diverse. And the flagship program that came out of the DAC is PEP. The PEP started in 2009, and every summer brings diverse undergraduate students to Woods Hole to do research for the summer. So this summer, we have our awesome 15 PEP students, and our coordinator who was a PEP student last year has come back to help out this summer. And so we're really excited to hear what you all have to say and see what you did this summer, and hopefully come back and join us for another experience or come back and work with us. So now I'd like to welcome the leader of the Woods Hole Diversity Initiative. The chair is currently Nicole Cabana from the NOAA Northeast Fisheries Science Center. Nicole is the deputy director, and she will welcome you.
Good morning. It's great to see you again. Thank you, students. Thank you, mentors. Thank you, supporters, everyone for being here. I don't know how many people we have online, but this is a big day. It was exciting for me as the Woods Hole Diversity Initiative co-chair to welcome you at the beginning of the summer. And now we are at the end, and we get to hear about everything that you've done, the research, the experiences that you've had. And it's always amazing to me how much you accomplished in such a short period of time. So I'm really excited to hear from each of you during the symposium today. I also want to say that this day is always a day that those of us who are affiliated with this program feel very, very proud. We knew that you had so much potential when we picked you, and we're proud of everything that you have accomplished and the experiences that you've had here this summer. And we thank you for choosing to spend your time with us. Any internship program is an opportunity to learn and discover. And I remember myself as an undergraduate, I did an internship program, and I learned a lot from that experience. A lot of things that I liked and things that I learned I wanted to do more of. And I also definitely learned things that I didn't like, and that helped steer my future path. And so I imagine you have had experiences like that this summer. And no matter what your experience was, whether you absolutely loved what you're doing and it confirmed that this is your interest and this is your journey, this is the direction you want to go in, Or if you learn, hey, this isn't maybe the thing that I thought it was, and I wanna do something different. All of that is really, really important and valuable and helpful to you as you figure out what your path will be. There's no one single right path, and you need to like really figure out what makes sense to you and follow that path, what the right set of decisions are for each of you individually. So I hope that for most of you, you had a fabulous summer, and that this confirmed that you want to continue careers and schooling, education, and science. And if you do, we would love to welcome you back, and there will be future opportunities to come back through the PEP 2 program that Angie's leading. And there will be many opportunities for future employment in Woods Hole, and I know that you learned a lot about those opportunities throughout the summer as part of your professional development. We want to continue to be resources to you, so feel free to reach out to anyone that you connected with this summer, myself included, and we would be more than happy to help you as you figure out what your next steps are. That's all I've got, and next I'm going to turn the mic over to Sylvester with the Marine Biological Lab.
Thank you, Nicole. Wonderful words. I agree with all of it. So thank you for that. So I am Sylvester. I'm the director of research here. And I can't believe that this is the end of the summer. I remember meeting you all. It seems like it was just yesterday that we welcomed you over at C Education. I almost feel that a time like this is a time to kind of reflect on what the experience was. And I'm sure you're doing that. I understand you're all leaving Sunday, so you've got a couple of days to do this reflection. I thought I'd share some thoughts about what I hope you take away with from this experience. What we all hope. I'm a huge supporter of the Woods Hole Diversity Initiative and this program in particular. And what we hope is that you go away with a great sense of accomplishment. It's just an amazing thing to come to a new place, a new institution, start afresh on a research project. You know that you have a kind of network here. So as has been said, several of you have come to the MBL. The MBL has many opportunities for you in the future in addition to the PEP 2 program. So please keep in touch with your network. Your peers are your greatest network. And so I know that this is one of the fantastic outcomes of this program. But the other important network is all of us. So we are open and we look forward to hearing from you in the future. So thanks very much. Have a great day. I'm really looking forward to hearing what you've done all this summer. So thanks a lot. Take care.
Now, the exciting part of the day, we get to hear all of you about all of the research and all the fun that you had at your presentation. When you come up to do your presentation, use the arrows on the computer to advance your slides. There's a laser pointer here that works very well. There is a timer on the presenter notes. When you get close to 13 minutes, if it's not obvious that you were on your final slide, you'll see either Autumn or I give you a nice wave. If you continue going too long, we'll stand up. But I know all of you has practiced so much that no one's going to go over the time, right? Yeah, exactly. So, a small, take a deep breath. Know that everybody, every single person in this room, every person who's online, is here to support you. We're here to see what you've done in the summer. We're here to support you in this experience, hopefully in your career, hopefully in everything that you do in the future. So just know as you look out on this audience, if you're nervous, if you forget something, if you think, oh, I should say this and not that, just know that all of us are here just to hear what you have to say. Whatever you tell us, we're going to be excited about. We know you all practice. We know you know what you did this summer. And we just want to be here to support you. So in case you haven't caught the theme, we are here to support you. We're absolutely here to support you. And so don't be scared, just know that you were talking among friends and people who want to see you. Okay? So that's the thing.
Good morning, everybody. Thanks for coming. My name is Calvin Payne. I'm a biology major at UMass Boston. I'm part of the Partnership Education Program, as you probably all know. I'm here working at Carolyn Teepolt's lab in the Woods Hole Oceanographic Institute. This summer, I don't know if you can hear me in the back. This summer, my research was focused on this question: has the European green crab benefited from parasite escape in North America? All right, so first I'm going to talk about parasites and their role in ecosystems. All right, sorry, I'm a little nervous, bear with me. So parasites are a healthy part of any ecosystem. They exert selective pressure on their host populations to increase their host defenses. They also control host populations, stimulating biodiversity within a biological community, and some parasites require multiple hosts to complete their life cycle, which helps them tie the ecological or the biological community together. For example, the trematode species Microphallus similis, which infects European green crabs. It's mature in the seagull, and then they defecate into the environment where it's picked up by periwinkle snails, who shed cercaria into the water column, and that is picked up by the European green crab, where it insists, and then the European green crab is eaten by seagulls, and the cycle continues. So invasive species are characterized by their prolific spread and their harmful effects on their biological communities. And parasite escape is one of the many factors that aid in that. Is one of the many factors that aid in their successful invasions. This cartoon is a figure trying to represent parasite escape. Parasite escape is when a species leaves their native range and is introduced into a new area where they escape their co-evolved parasites. This happens one of two ways: either they take the parasites with them and they don't have any of the other hosts within their life cycle to complete the life cycle of that parasite, or they just don't take any parasites with them and are happy to escape and chill at the beach. My slides over. All right. So the invasive species I'm going to be talking about today is the European green crab, or Carcinus spinus. They were first detected in Massachusetts over 200 years ago, and that same population spread all the way up to Newfoundland. The same population was then introduced again on the West Coast in 1989, and it spread all the way north to Alaska. And they have basically, because of that secondary introduction, they have benefited from parasite escape not once, but twice. European green crabs are terrible on eelgrass beds, which are a nursery for many aquatic species. They're also interfering with softshell clam fisheries as well as mussel fisheries, and they're outcompeting juvenile Dungeness crabs on the West Coast. So my major question is, does parasite escape contribute to green crab success as an invasive species? So like I said, we know that they may have benefited from parasite escape the first time coming over to the East Coast. That was over 200 years ago, so it's likely that they have parasites now, as some of their native parasites may have migrated over, or some of the local indigenous parasites have evolved to infect them. On the West Coast, having gone from parasite escape twice, we believe them to be parasite-free, at least previous research has shown it. So the first question I had to ask was, what are the parasite loads and prevalences in meat? And does parasite escape give green crabs a physiological benefit in dealing with secondary stress like heat? The arrows don't work, by the way, I keep pressing. All right. So to answer the first question, look at parasite load and prevalence in New England. I collected specimens, I set up traps all along Cape Cod. And then I dissected those crabs for their digestive organs and their digestive tract to identify any possible parasite infections. Using microscopy, I was able to identify. So the first parasite that I found was a metacardial cyst, which you can see in this first image. Individual cyst, what happens is the same trematode species I talked about before that requires multiple hosts to complete their life cycle. The crab is one of their intermediate hosts. And when the parasite gets into the crab, it insists into their hepatopancreas and forms one of those. A high parasite load of metacardial cyst looks like this second picture here. The second parasite I identified was an acanthocephalin. Similar to the trematode, this one is a spiny-headed worm, and it also requires multiple hosts to complete its life cycle. In the crab, again, an intermediate host for this parasite. It forms a cysticanth inside their digestive tract, which you can see in that darker oval. The arrows. Also, again, a picture of what high parasite load might look like with this parasite. All right, so I was able to survey 5 different populations of crabs. First, I started here in Eel Pond, right outside, with a population that I found to be parasite-free. Now, this map shows all the different locations and the prevalences are in pie charts. So the brown color represents none, which is what I found in Eel Pond. And then I continued to travel along Cape Cod and was fairly unsuccessful in catching crabs until finally I went all the way up to Chatham and I caught some crabs. This population was 100% prevalence of acanthocephalins, which is represented here in the yellow. And then I was also able to receive crabs from these other 3 locations. And the Quahog Bay Conservancy sent me crabs from Harpswell, Maine, which had a high prevalence of metacardial cysts, which you can see here in the orange, but a relatively low parasite load. And then second, I got from Newcastle, New Hampshire. My PI was kind enough to bring them back for me. They had both parasites present, which you see represented in the gray, and then also, obviously, some had just acanths and metacardial cysts as well. And my last population that I surveyed was from Willapa Bay, Washington. Here, like I said before, they possibly benefited from parasite escape twice, and I did find no parasites in this population. So now that I have a good picture of what the parasite load and prevalences are, I decided to use these 3 populations represented by the star in my next round of experiments. I use these populations because Washington ones, being parasite-free, are a good representative of my parasite escape population. And then with New Hampshire and Maine populations, they give me a good variable range of parasite load and parasite prevalences. With using the buttons. All right. So my second question, does parasite escape allow green crabs to better tolerate additional stress? So I answered this question by measuring their time to right. And time to right is how long does it take for a crab on its back to flip over onto its legs? This is a good physiological measurement of their health, really. So what I did for the additional stress was I subjected them to 30 degrees Celsius for 22 hours. I took their initial time to right beforehand, and then again at 22 hours into the 30 degrees Celsius. And I expected to see that if they were not tolerating their parasite loads, or maybe they had no parasite loads, either way, I expected to see a difference in their time to right from before and after. I expected that to be a longer time frame for them to be able to flip over. Yeah. All right. So in that experiment, I was able to plot some data. I got your average time to write on the y-axis and my 2 time points on the x-axis being their initial time point and then 22 being their 22 hours, 30 degrees Celsius for all 3 populations. I found a significant difference in their time to write in both the Maine and Washington populations. However, in the New Hampshire populations, it was not significant, but I think this is probably because my sample size was only like 15 crabs and they had a very variable response. But what I'd like to draw your attention to is that the Washington crabs did have a significant change, which means that this was a heat stress event and it did actually stress the crabs out. All right, so this graph is a scatter plot representing parasite load versus their change in time to rise. So this is just the difference from their initial to their second time points. And I measured it with parasite load. So again, y-axis is change in time to rise, x-axis parasite load. The locations depicted by color here, blue being Maine, red being New Hampshire, and the different shapes are just the types of infections that they have. The circles obviously being anencephalins, the squares being metasecarial cysts, triangles representing both, and a plus sign for none. I expected here to see an increase in slope as parasite load went up. I expected the change in time to the right to also go up. As you can see, that did not happen. Turns out these crabs are very resilient and handle a large parasite load. For example, my furthest point to the right here had over 400 metasecarial cysts in its body cavity, and it increased its speed and got faster after the liked it. I don't know. And then you can also see that the top point, that red square up top, had maybe less than 20 parasites, and for some reason it took longer than everybody else. So clearly they tolerate a pretty wide parasite. This next slide, I added the Washington data if the slides will cooperate. Here we go. All right, so these little green plus signs, maybe you can see them in the back, I'm not sure, but basically what I'm trying to show here is that they had a very variable response as well, similar to my Maine and New Hampshire populations, despite them being completely parasite free. All right, in conclusion, I believe these green crabs can tolerate a wide parasite load. The reason for that being that in their home range, they're subjected to many different parasites that co-evolve with them, as I said before. And here I was only able to find 2 of the most common ones. Some of the parasites they deal with back home are viruses, rhizus, I don't remember the name, to be perfectly honest, but like a castrated parasite that hits their time here. And so I think that further research would obviously be needed here. The green crabs are a very robust host defense to parasites. In the future, I would love to repeat this study for the full heat wave of 5 days. It's something I didn't really have the time to do this summer. I'd also like to genotype the crabs for further analysis. In my lab, the previous research has shown that there's a region of the genome that houses most of their temperature genes, temperature-related genes. And because of that, they have 2 alleles, one more cold tolerant and one more warm tolerant. And I think that that might better explain some of my unexplainable results here. Oh, I guess I kind of missed a couple. But anyways, I'd like to include native populations from Europe as well, represent the actual native range of parasites for these species. And I'd like to compare parasite load and diversity with some of the indigenous crab species to kind of see how much they're actually benefiting from parasite escape. And I'd like to thank Dr. Carolyn Teeple and Dr. Yami Venkataraman and all the PEP interns and my lab mates that helped me flipping crabs this summer. And of course, the PEP organization for allowing me to be here and for this great opportunity to do research. With that, I will take any questions. Yes.
Great talk and very clear experiment. Hope you had fun with it. I was wondering, is there any other metric other than the time to write to assess the impact on their physiology or success?
Yes, we have at least one that I can talk on. We can measure their heart rate. There's something I was unable to do, just time constraints. But we could have taken a measurement pre and post of their heart rate and see how their tolerance is.
Great, thanks.
All right. Did you get pinched by any of these crabs?
Yeah, in particular, there was one called number 66. He got me through the glove. I had to drive the glove on, and he still got in there and broke skin, actually. Really nice. Oh yeah, these are some of the catches that I've made across. All right, well, thank you very much.
Hi everyone, my name is Maddie Stewart and today I will be talking about my project this summer, which was modeling entanglement risk and risk reduction measures for Atlantic large whales. Okay. Like hard. Sorry. So this summer I was an intern in the NOAA Northeast Fisheries Science Center in the Protected Species Division, and I worked with a team who is currently trying to model entanglement risk for Atlantic large whales. And my project focused on conservation measures about minke whales, humpback whales, fin whales, and right whales. So humpback whales and fin whales are being studied by the team, but North Atlantic right whales are the highest priority right now. There's around 360 individuals left of the population, with about 70 reproductively active females. This makes them the most endangered large whale in the Atlantic. They're extremely susceptible to commercial fishing gear due to their extremely arched jaws and really rotund bodies. Their populations were decimated by the colonial whaling industry, and they haven't bounced back quite as well as humpback whales or fin whales. Okay, so what do we know about dangers to right whales and how to mitigate them? Entanglement fishing gear can be fatal to right whales. Johnson et al. 2005 found that right whales were most commonly entangled with their jaws and around their body, and humpback whales are most commonly entangled around their flukes. Okay. So right whales are most commonly entangled by pot and gill net gear, which is also known as fixed fishing gear, and that's the most common commercial fishing gear used on the East Coast for lobster and crab fisheries. And knowledge of species distribution is incredibly essential to understanding how we can conserve these animals. If we know where they are, where they're getting entangled, and why they're getting entangled, we can create conservation measures to help protect them and help the population bounce back. So the effects of humans on right whales became extremely apparent in 2017 when NOAA Fisheries declared an unusual mortality event for this population. An unusual mortality event is an unexpected stranding or large die-off of any marine mammal population, and it requires an immediate response. So this UME resulted in elevated mortalities of North Atlantic right whales in the US and Canada. And the 6 dead and injured whales consisted of about 20% of the population at the time, which is really significant part. The leading cause of this unusual mortality event was anthropogenic, primarily entanglement in commercial fishing gear and vessel strikes. And so if you see this bar graph in the corner, you can see starting in 2017 how birth rates of North Atlantic right whales really plummeted while the death rates increased. So you can see how detrimental this was to their populations. Unfortunately, right whales are not the only Atlantic large whale going through an unusual mortality event. In 2016, NOAA Fisheries also declared humpback whales going through an unusual mortality event. And the causes of this are really similar to that of the right whales: entanglement, commercial fishing gear, and vessel strikes. So you can see in this plot in 2016 how annual humpback whale strandings from Maine to Florida increase. And both of these unusual mortality events are ongoing, so they haven't stopped. So in response to both of these UMEs, the Atlantic Large Whale Take Reduction Team amended their take reduction plan in 2021 to help mitigate these issues and to primarily help the right whales. So they implemented some new time area closures that you see outlined here in yellow. You have an extension of the pre-existing Massachusetts restricted area, so it goes a little bit north. You have a new lobster management area called LMA1 restricted area, and you have the South Island restricted area, which is just south of the Cape. They also implemented new trap per trawl requirements. So this plot shows the New England coast, and each color correlates to a minimum number of traps per trawls for those lobster fisheries. And a typical trawl has however many traps, but they have 2 single end lines. And this is very dangerous for right whales because that's how they become entangled in commercial fishing gear. So instating a minimum number of traps per trawls helps fishers fish the same amount of traps, but with less single end lines in the water. So this brings me to my research question, and this summer I've been thinking a lot about conservation actions and how much effort is going into saving right whales, but we don't know how much our actions for right whales are affecting the other species of large whales in the Atlantic. So I was wondering, how do risk reduction actions for right whales affect humpback, fin, and minke whales? So my goal was to plot the seasonal distribution of each whale, find out where they most overlap, and then try to come up with conservation measures that can benefit all 4 species. So before I get into my methods, I wanted to talk to you all about an example of one conservation action that was in place just to benefit right whales. So this was that South Island restricted area that I was talking about that was implemented in 2021. You can see it's just south of the Cape. This conservation action was implemented because this is an area where there was a lot of trap pot gear with that single end line. So this figure shows how easily a right whale can become entangled in this commercial fishing gear. That single line takes up the entire water column, so it's kind of impossible for a right whale not to run into it when they're foraging. The issue with this area is that it's still open to gill net gear, which does not pose a large threat to right whales, but it is a huge threat to humpback whales. When they dive down deep to forage, they will run into the gill nets that are sitting on the substrate and become entangled in that. So I am thinking about how we can reconcile making these safe spaces for right whales while still putting these other species in danger. So in order to do all of my plotting and modeling, I used NOAA Fisheries Decision Support Tool, which was a tool developed to help managers and scientists understand seasonal distribution of whales and risk entanglement and threats. So this model combines the fixed gear fishing layer, which is what I was talking to you about with the fixed gear with the pot and gill net gear, on top of a species habitat model that plots seasonal distribution of these 4 species of whales. And that results in co-occurrence, which is the co-occurrence of a whale species with fixed fishing gear on the northeastern coast. So using the DST, or the Decision Support Tool, I decided on 2 management actions that I think could benefit all species of large whales. So the first management action was requiring single end lines in all the federal waters of LMA1, which is that blue area right there. So this action will help reduce the amount of single end lines in the water with trap pot gear to help reduce entanglements for all species of whales, but primarily right whales. A typical trawl, again, has 2 end lines, and both of those end lines help fishers understand what direction the trawl is going. So when they're out getting their gear or they see other fishers' gear, they know where it's going. So they normally have 2 end lines because in case one breaks, they have an extra one to help pull it back up onto their boat. But with this single end line measure, fishers can either decide to just keep one end line and kind of rely on that one, or use a ropeless device on the other end. And this action can help reduce end lines in the water, but it also incentivizes fishers to start using more ropeless gear. The second management action I decided on doing was a lobster fishery 400 pot gear cap in that same LMA1 area. So the current gear cap is around 800 pots per fisher, but not all fishers fish that whole 800 amount. What was really interesting was learning about the differences between the Canadian and American lobster fisheries. So in this 2020 Myers and Moore paper, they found that the US lobster fishery in Maine expends approximately 7.5 times as much effort as the Canadian fishery. However, the Canadian fishers catch about 3.7 times more lobster per trap. So not only is the US lobster fishery incredibly inefficient, it just also has a lot more gear in the water, which poses a greater threat to whales. So with this 400 pot gear cap, it's not only more safe for the whales, but it's more economically feasible for the fishers. They're spending less time on the water deploying and getting their gear back, and they're spending less money on bait, which is expensive. So here are some of my first plots. So these are plotting the baseline co-occurrence for all 4 species of whales. So on this legend, you see that the green, red, and orange colors are areas of higher co-occurrence of whales and fishing gear. This red area, the purple area up north, is that LMA1 area that I showed you earlier, and this green box below is that South Island restricted area. So you see areas of high co-occurrence in and around LMA1, and that is that South Island restricted area. So because there was so much co-occurrence in that LMA1 area, I decided to focus all of my management actions. So this was that first scenario, the single end lines in the federal waters of LMA1. The legend for this is a little bit different. It is the difference in co-occurrence between baseline co-occurrence and the new co-occurrence of this management action. So there was a 6% reduction in end lines, which means that 6% of the gear was removed from the water. And this resulted in right whale co-occurrence being reduced by 17%. That is a pretty big number, and the other whales also benefited. So with fin whales reducing co-occurrence by 10%, minke whales at 8.5%, and humpback whales at 11.4%. So right whales had the biggest reduction in co-occurrence, and because all of these numbers are bigger than 6%, this means that all of these whales are disproportionately protected by this management action, which is a really good thing. So these are the plots for my second scenario, that 400 pot gear cap. And that legend is the same. It is the change in co-occurrence from baseline co-occurrence to co-occurrence for this management action. So I saw a 27.5% reduction of gear coastwide. So along this entire LMA1 area, around a quarter of the gear was reduced. And this resulted in humpback whale co-occurrence being reduced by 28.7%. And the rest of the species had approximately 1-to-1 ratio of gear reduced and co-occurrence reduced. So some of my takeaways and conclusions: that single end line action benefited North Atlantic right whales the most, and the risk to whales was disproportionately reduced relative to how effective the fishery was. So this is a really, really great thing. That 400 pot gear cap, there was less risk reduction than I anticipated, which means that this action might work better on a smaller scale rather than coastwide. So if you restrict it to just one state's coastline or just a smaller fishery, I think there could be more risk reduction. And for those closures, that South Island restricted area benefited right whales tremendously, but not so much for other species. So it's really important to think about, does the gear moved outside of these closures pose new risks to other species? These are some of my references and my acknowledgements. I think my whole team is on Zoom, so I just want to say thank you to my mentor, Mike Asaro, Laura Solinger, and Alicia Miller, who helped me create all of these plots. Obviously, the NOAA Northeast Fisheries Science Center for giving me an incredible opportunity, and
Do you know anything about whether the filters themselves would be more tolerant of some types of decisions rather than others?
yeah, so that's the great thing about working with individual support tools, that you're not only working with scientists, but you're also working with fishers and managers and industry. I think that in the beginning, a lot of fishers would be kind of pushing back on changing the way of fishing, especially with the lobster fisheries, because that's been done the same way for so long. But I think once you present it as they'll be spending less money on bait, less money for gas for their boats and everything, I think they will come around to it. Yeah, I think it'll just take a little bit of time.
Another question from the chat. Your recommendations were to be implemented. Do you have estimates on how much the right whale population would recover in terms of growth in the coming years?
Yeah, so right whales are so sensitive to any anthropogenic effect. I think it would be hard to say exactly how much they might grow. I think a significant reduction in single end lines coast-wide across the entire East Coast would definitely help their populations. But yeah, I mean, it's really hard to say. They are a very sensitive animal, so that action could help them, but then, you know, they could get hit by a boat and it would not be good for them. So, yeah. Thank you.
Morning, everyone. As it shows up, I was part of the PEP program for the summer, and over the summer I was able to work over at SGS with Eagle. And the name of my project was Enhancing Coastal Stability Through Restoration Projects in the Gulf of Mexico. So, the environment that I worked on was the coastal dunes. So coastal dunes are basically just big mounds of sand that build up over time, and they originate from small foundations like plants or rocks. And eventually, when wind is picking up the sand, it gets caught up on these plants and these rocks. And eventually, when they build over time, let's pretend my right elbow over here is a beach. My left is the mainland. This will basically build up, and the structure will block off wind that's being pushed in from the beach. So we will go, and we'll basically miss anything that's down here. So it will allow for like this succession of vegetation. So it could be small plants, bushes, shrubs, and eventually they turn into big trees. And a bunch of animals use this environment, as there are many uses for it. Where you find a bunch of fruits on these plants. For example, we'll get into that. People more than food, you can find shelter in these areas, and also in these areas. And these systems are pretty sensitive to anthropogenic effects in the Gulf of Mexico. Okay, so in the Gulf of Mexico, this is where I focused my project, and you can see from Texas to Florida is where I did my study. And on this bottom right graph, you can see it's all red. So on this legend over here, you can see it turns red, it turns all the way blue. Blue means a colder temperature, and red means a warmer temperature. So does anyone see any green, blue, yellow? No. It's a large system. It's a rather warm system. I consider it kind of like a large crock pot, slow cooking, basically going to feed into producing a lot of storms, and we'll get into that. And so the involved managements that I worked with over the summer were U.S. Fish and Wildlife Service, Florida Department of Environmental Protection, National Park Service, and the Florida Gulf Islands National Institute. So some of the challenges that are present in the Gulf of Mexico are the Deepwater Horizon spill, which is basically in 2010, NOAA rig released tons and tons of oil and affected the entire Gulf Coast of the Gulf of Mexico. And a bunch of systems were hurt by these, for example, beaches, lagoons, and marshes. And and climate change has also been affecting the Gulf of Mexico. So over here in this picture, in Perdido Key, Florida, there was a bunch of rain during just one storm. And you can see these cars are basically totaled from these waters. And in the Gulf of Mexico, there are also the introductions of invasive vegetation. So down here on the bottom right, torpedo grass is connected by the same root systems, but it's going to have different shoots. And basically, it'll be able to spread and encroach on native vegetation, basically kicking out native vegetation and becoming a dominant species. And basically, these shallow root systems, so you can see they kind of just look like, I don't know, they wouldn't seem like they would hold in too much foundation on the floor. And basically, this can lead to the downfall of dunes. And a bunch of animals, like snowy plover and the beach mice, use the Gulf of Mexico to live. And for example, snowy plovers use these locations to, as they migrate, from the latitudes and basically use this place mostly to forage and build up their nutrients as they go up north. And beach mice well, both snow plovers and beach mice are both threatened. And for example, dune mice are going to stay within these dunes. And basically a bunch of these vegetation has been lost. Like for example, this torpedo grass is not going to allow for much shelter for them to hide in, and also not forage as much since they don't provide food sources. So in an effort to combat these effects on the Gulf of Mexico, restoration projects are done. And some of these restoration projects can consist of sand fences. So down here on this left-hand side, you'll see in Louisiana, seems like the sand fences under all the sand, that's exactly what it's doing. It's building up this sand to basically at some point just keep going up and up and up. And then also planting vegetation. So on the bottom right side over here, you can see a bunch of seal was planted on the Gulf Coast of Florida to improve just dune retention of sand. And then on the top right side, you'll see here in this McFadden National Refuge in Texas, they're dumping a bunch of dredged sand onto the coast, attacking a marsh system that is just back here. So the location of my projects, this ranged from Texas to Florida, like I said. In Texas, there was around 7, in Alabama, there was 3, Louisiana 6, and Florida 7. So the question for my project was, are these restoration projects replicating a natural dune system where it can retain proper dune height? So for my hypothesis, restoration projects will help to retain dune height. So in the northern panhandle of Florida, right over here, you can see this is where my research location is, which is Perdido Key, Florida. Just on this part right here is where we're going to focus on, is where a project occurred. You can see on this timeline right here that in April 20th, 2018, the Perdido Key dune restoration is here done, and then in September 2020, Hurricane Sally on this bottom left image hit directly into Florida at a category 2. And on this timeline, you can also see that in April 2017, November 2018, and October 2020, DEM images were reported, which are digital elevation models. And from there, yeah, so then from here, the project consisted of 5 miles and 20 acres of area covered, around 20 to 60 feet inward. So just off the coast, like literally, like we see on the map, you'll see that it's right next to the water line. 1.05 million cubic yards of sand was placed in, and then also planted native vegetation. And this was done in April 2018, like I said. And the vegetation that was planted consisted of sea purslane, beech elder, sea oak, and cordgrass. And on the graph on the top left side, you can see some of these plants are represented. So for example, sea oats and bitter panicum, which is panicgrass, and cordgrass were planted on the actual dunes themselves, and they're going to be a lot taller. So you will notice that this is pretty lanky, somewhat taller compared to these two, sea purslane and beech elder. So sea oats and cordgrass are specializing in retaining the sand that's being pushed from the wind, and behind that you're going to see this sea purslane and possibly beech elder as well. So they're going to be a lot lower in height. They're basically going to flat out these dune areas that follow afterwards. Will basically provide foundation for this to kind of just stand up along with this vegetation on top. So for my methodology, I used ArcGIS Pro, and I was able to download images off of the NOAA Data Access Viewer, and I was able to get DEM images, and from there I was able to compare them and combine them together, and then from there infer the success of these restoration projects. So this is 2017. This legend shows that the red, so the red right here is gonna it's gonna say that it's a lot taller, and then green is gonna be a lot shorter. This is for 2018. This is for 2020. And then using these images, I combine them together. So for example, 2017 and 2018, you see there isn't much difference, but once we zoom in, you can see that along the entire coast, there is an increase in height. So this is when the restoration project occurred. And then we can see back here that there's a decrease in height, but this could be from the actual environment stabilizing and kind of adjusting to this new input. And so then the same thing occurred in 2018, 2020. But this in 2020, if you recall, Hurricane Sally had directly hit Perdido Key. And if we zoom in again on the eastern portion, you can see a bunch of this actual coast was degraded following behind the coast, you can see that there's a bunch of blue. So sand wasn't necessarily lost, it was more so just displaced. And on the western portion, you can somewhat see the same thing. So it wasn't necessarily lost, it was kept within the system. And here in the vegetation images, so just normal imagery, you can see that in 2017, it's somewhat bright, can't really see too many shadows. These shadows are gonna represent vegetation itself. And in 2018, you can see it's a lot darker, following the restoration project that occurred. In 2020, like I said, there was a hurricane. We can see that a bunch of water in the actual lagoon system. This could occur from just a bunch of rain that have happened, but also overwash events, which heavily impact sand erosion. They have water itself has more power than wind does. And here in 2021, you can also see that the prevalence of plants over here more than 2017. So it was able to basically sustain these damages from the hurricane itself and kind of just continue the foundation on the dunes. So based off these results, my hypothesis was accepted. Dune height was preserved following a restoration project. The dunes kept proper composure, which could allow for success in a coastal dune ecosystem. And then also vegetation can be inferred as one of the reasons why the dune heights were able to persist. They didn't use sand fences, but native vegetation to basically impose an increase in height and kind of sustain these environments as they were. So some of the conclusions I reached through this project was that management services have to take into account many of their needs. So like I said in the beginning, there was at least 5 organizations that we worked with, and each one has a different focus. So for example, here we can see this beautiful little snowy plover, it's very cute. The snowy plovers are dependent off of these systems at stopover sites. So different management services are going to focus on snowy plover, others on the beach mice, others on the protection of the actual coast, and then others on humans. And for protection, I wanted to make sure that you guys realize that these are first line of defense. In the Gulf of Mexico, a bunch of these hurricanes are more intense and in increased numbers. And this is actually our first line of defense, like I said, but also this is the home for a bunch of these animals. And we need to take care of these animals as this will promote biodiversity, but also improve ecological services and continue to allow these ecosystems to function. So as of recently in May 2022, the same project was done, just inputting dredge sand into the system. And these are some of the efforts that are continuing, not just through Perdido Key, but also just through the entire Gulf Coast. And I want you guys to think a little bit bigger beyond this project. So here is an image of all the outlines of the actual coast that I took from 2017, 2018, 2020. And you can see they're kind of staggered on top of each other, but the reason is that these coastlines are moving. And these coastlines, this key is basically moving, I don't know exactly how many feet per year, but this is gonna always move, and these systems are always gonna require some type of attention and making sure that they receive the proper care. But also that because these coasts are the first ones to receive the impacts of storms, they will protect us, but also you need to take into consideration where you live and also where these animals live. Economically, these projects mitigate habitat and infrastructure damage, but also promote ecotourism. And mainly because these dune ecosystems or these coastal dune ecosystems are not only in Gulf of Mexico, but just very unique places like the Gulf of Mexico. And ultimately, we want to save the integrity and purity of this planet as we go on. And these are my references. These are the institutions we worked with over the summer. And I just want to say that in future years, NOAA is one of my positions I want to fill in. And I just want to say thank you very much.
Not a question?
Yeah, hi, thanks, that was great. So I'm gonna do the thinking bigger part of this, and I'm a native of Florida myself. You're in school on the on the open water side, on the Atlantic side. And I'm wondering about the barrier islands there and how you think, I mean, they're so much more exposed, right? And my own sort of experience with the dunes on that coast, which is where I also grew up, is that they're just constantly needing sort of re-nourishment. Do you think is there any hope to sort of, with sea level rise and with that sort of exposed coast to sort of use these methods on that side of the state?
Yeah, so on the eastern side, I guess it's gonna be, the Atlantic Ocean. There aren't as many barrier systems. There is the Indian River Lagoon, which is gonna somewhat be like a barrier system, and it's well connected. But like I said, the Gulf of Mexico is just a really intense pot of just warm water that's just gonna feed all these storms, and it's gonna increase the amount of storms, the severity, and also just like wave action as well from these storms. So wave actions, I would say, are probably the most prominent issues to just coastal erosion. It's going to be the most powerful, and eventually this overwash just keeps reaching over and over and over, and slowly the sand keeps being displaced. But yeah, it's I would say more in the Gulf of Mexico, it's I guess needed. Most of these dune ecosystems are in the Gulf on the Gulf Coast. But yeah, it's a matter of time when we see these changes. And, let's see what efforts we put in. Any other questions?
Hi, everybody. My name is Sylvester, and over the summer, I have been working with Dr. Laura Milano to study inactive sulfide bed communities. But before I explain exactly what an inactive sulfide bed is, I want to give you a little background about active hydrothermal beds. So what is a hydrothermal bed? Well, vents form when seawater seeps into the crust. And when it's there, it gets heated up to really high temperatures by all of that magma. It takes in minerals from the rock around it, and it rises again. And then when that superheated hydrothermal fluid meets the really cold, that causes all of those minerals that were in the water to be deposited. And that's how you get those really interesting looking spires and mounds, things like that. These vents are home to thermophilic or heat-loving bacteria. These bacteria are also chemolytic. While plants draw energy from sunlight, these bacteria draw energy from chemical reactions and inorganic chemicals, like the sulfur found at these vents. And in the deep sea where there isn't a lot of sunlight, there aren't a lot of plants, these bacteria become really, really valuable parts of the food webs in these ecosystems. So that means that hydrothermal vents become biomass hotspots in the deep sea. So what are inactive sulfides? Well, they're the mineral deposits left over after that hydrothermal activity wanes. And this happens, as the sulfide deposits move away from that heat source, usually at spreading zones where two tectonic plates are moving apart from each other. And without those hydrothermal fluids to support thermophilic bacteria, you might assume that these deposits are, barren or devoid of life. However, as that hydrothermal activity wanes, thermophilic bacteria are replaced by bacteria that are more suited to the cold. And those bacteria can still use those same minerals that are there to produce energy, even without the heat source. And they can still be a really good food source for the animals living there. And as it turns out, inactive sulfides might even be more diverse than active vents. And there are many gastropod species that are thought to be specific to inactive sulfides, which basically means that they are specifically adapted to live and reproduce in those environments. Gastropods include limpets and there. And something that is really cool is that the two snail species you see on the right are completely new to science. Before they were discovered on inactive vents, they had not been discovered anywhere else. You might be wondering, why should we care? Why does this matter to us? As I've said before, hydrothermal vents are full of really valuable minerals. And those minerals are really attractive to the up and coming deep sea mining industry. But the thing is that active vents can be really dangerous for deep sea mining operations. If that equipment comes into contact with the superheated water coming out of the vents, it could really damage the equipment, which, companies wouldn't want to risk. Inactive sulfides, which lack that danger, seem like a really good alternative. But the thing is, research on inactive sulfides is decades behind the research on active vents. So that means we just don't know enough about the communities on inactive sulfides. We don't know how reliant they are on those environments or how connected they are to the rest of the ocean. And we don't know how mining would affect the animals living there. And that's why our lab is studying these inactive sulfides to get a better idea of the communities that live there and how they work. So, my project, I used 5 samples collected from a feature called Loftus Mound on the East Pacific Rise, or the EPR. Those samples were collected at a depth of 2,500 meters. And you can see on the left is a map of the EPR, and on the right is a zoomed in look at our study site where samples were collected. These samples were collected back in January using the submersible Alvin, which you can see there. And since then, those samples were preserved in ethanol until they were ready to be sorted. So we wanted to explore whether the animals on these inactive sulfides displayed any habitat preferences between the different rock types. So each of our 5 samples was taken from a different type of rock. You have the rusty yellow rock at the top, the rusty orange rock, the brown rock, and the green kind of rock. And we also had some transitional rock types, which are sort of in between 2 of the colors. And once we had those samples, we had to, take the animals out of it, and we really needed a microscope to find them within the sample, because a lot of the time, the animals that we're looking for are only a couple of millimeters long, or sometimes even shorter. And after we found those animals, we identified them, we counted them, and we sorted each species into their own line. In addition to sample sorting, I also did a little bit of image analysis. So to do that, I combed through the videos from the dive where the samples were collected. Then, I found images that I thought were representative of each rock type that I wanted to study. And once I had those images, I randomized a 15 centimeter by 15 centimeter quadrant within each image. I cropped it down to that, and then I counted the number of snails within that square area. And all of the little white dots that you see within that image are snails. So it took a while. Once we had that number, we could divide the number of snails by the total area to get population densities. And this is the result of that. So as you can see, the one in the middle, which is Rusty 3-4, is sort of in between the orange and brown rock types, had the highest density, and brown 4 had the lowest density. So there was a lot of variation, but because we don't really have a lot of replication for these, it's difficult to tell yet whether this is actually due to the rock type. And this is the results of our sampling. So the bar on the left, which is Rusty 2, the yellow rock type. It has sort of low diversity. It's really dominated by and the one next to it, Rusty 2-3, sorry, is missing one species that all of the other rock types had. But the thing is, that might just be due to the fact that by the time I was able to sort that sample, some of the gastropods had started to dissolve, and we aren't really sure why, but it did affect our ability to identify some of them. And like with the population density, there is a lot of variation, but it's really hard to tell whether that is because of the rock type. We would need more sampling and more analysis to really get a better idea of these populations. So in conclusion, we saw some hints of species association with rock type, but there wasn't really any clear pattern. There's a lot of variation, and with more research that could prove to be ecologically important, but again, more sampling and more analysis is needed. What we do see is that the gastropod species collected here appear to be specific and endemic to inactive sulfide events. Like I said, some of these species were unknown to science before they were discovered on these inactive events. And anyway, these communities are novel to science. We have a lot to learn about them. And because they're potentially so vulnerable to mining, we need to know more about them before we make any decisions about, utilizing them for ourselves. And my hope is that the work our lab is doing will encourage more research into these little studied ecosystems. And these are my acknowledgements. I want to thank everybody in my lab and the Alvin crew and the Sentry crew, as well as the NSF. And a big thank you to everybody who's a part of PEP and CUI.
Wondering if you know anything more about either the mineral composition of the rocks or the microbial communities on them that might account for some of the differences you saw.
I think my project didn't really cover that, but I know that there are people working with my mentor to study things like that, so I think we're working on that.
Good question from the chat. Could inactive sulfide deposits help scientists understand other planets?
I think so. I know that there are similar environments in other areas and on other planets, so I'm assuming that it would be possible to connect the studies of these environments. My project didn't really encompass that, so.
Do you have a favorite gastropod this summer?
One I found a lot of was the Portland, the top right there.
Hello, my name is Jordan Barrett. I go to the University of New Orleans. I'm a chair here. Well, I'm a research student this summer with the student data. And my focus this summer is undergrad analysis of bound and surrounding areas. So climate change is disrupting weather patterns, causing warmer temperatures, increasing precipitation patterns on our winters. And the Northeast is warming faster than other regions due to the Atlantic oceanic current. And as you can see in the graph right there, red is like warmer temperatures, and everything that goes lower is lower temperatures. And Falmouth and like other areas, Cabana, Massachusetts, it's areas. that can be linked to water quality because, algae growth, tend to be faster over temperatures. And with, the focus on increased temperatures, that can increase water runoff of nutrients and like nutrients like nitrogen and phosphorus usually cause like those dissolved oxygen rates to low. And this could also, like warmer water can also lead to like stronger storm intensity. So my research question is, we will work these regions faster than other regions. Looking at a Fahrenheit scale, it is found that warming faster than surrounding locations. The methods I applied were targeting daily precipitation and temperature data from 1992 with a high coverage, because anything with a low coverage means there's gaps in the data. I found records for Falmouth through the town of Falmouth from Doc Taylor, Richard Payne, and for New Bedford, Plymouth, and Taunton. I used NOAA's climate data online tool. I used RStudio to separate each location by year, season, and month. I calculated slope P values to see how much temperature or precipitation is changing. I looked for trends within those temperature and precipitation values, and then I compared those locations at the location. So for here at this first graph, we have the yearly average maximum temperature with trend lines with all 4 locations. The green line is Falmouth, the orange line is New Bedford, the purple line is Plymouth, and the pink is Taunton. The reason there is a big, like the green line is long, or Falmouth is longer than the other locations, maybe it's because I got public records of Falmouth through the town of Falmouth, and everything else was online, so the coverage is like fairly low for anything that went further back than 1992. And if you look at the P values, they are and if you like look at the slopes, they are close to 1 degree Fahrenheit each year, and like the trend line shown that each one is increasing, but this also shows that Falmouth is not increasing more than other areas. And here for because my focus is on Falmouth, I wanted to see I wanted to compare each season. So this one has the summer months of June, July, and August with a focus on Falmouth. I wanted to see which month in the summer had the largest slope, and for this one, it is August with around 1 degree, or 0.1 degree every year. And to put that into like, you know, I guess like larger terms, like that is around 1 degree Fahrenheit every 8 years in August. So I wanted to take a focus on the month of August and compare it to other locations. So I did August, month of August for all the other locations, and they are all increasing. But according to the slope, often have the largest increase, and the P values are fairly low, so that just means that there's good significance in that result. So next I took Falmouth and the winter months of December, January, and February, and did the same thing. I saw that there was a larger slope in January, so I compared those. I compared the January months with the other locations, and the same thing. They all have increasing values, but Taunton has the largest increase, and they all have really low P values. And so, with that wrapping up temperature, I went ahead and looked at total precipitation to see if there was anything significant, and there is an increase of precipitation for Falmouth, but the other locations are pretty variable. One of them, New Bedford in particular, shows a decrease. And the p-values are not as small as the ones before, so there's like I guess like it's not as like significant. And so again, I compared the summer months for each location, and there's not a lot of significance along with winter. And so my conclusions are that warming is occurring rapidly across southern Massachusetts, more so in the summer than winter. But that is inconclusive whether Falmouth is warming faster than surrounding locations. So basically that's just agreeing with like the article that I mentioned earlier, that it is warming at a regional scale. Warming is consistent with ocean warming caused by the Atlantic meridional or returning circulation, which is slowing in the warm water from the Gulf Stream is heating the region. The precipitation change was not significant. And so the implications for water quality, the summer warming is a big deal because of the warming temperatures, and you know, there's not a significance in the increase in precipitation. Temperature is likely a bigger issue for water quality than precipitation patterns. So the next steps I would say to take would be to calculate minimum temperatures for winter to see if there's a warming trend for the lower temperatures, to see if you know if it is actually getting warmer, because the maximum temperatures just show like the highest temperature. But further data analysis can look into potential storm intensity. And these are my acknowledgements. Thank you, Dr. Neal, for for mentoring me this summer, and Dr. Kristen Heisinger for all the field work experience and helping me with all the R coding. Drew Lehman, he is a mentor I had a couple years ago for a different internship, and he introduced me to a lot of different like opportunities, including PEP. Anji, she is the director of PEP, and she was actually a guest speaker for the other internship I had, and she's just always been there for us and always made sure that we were like feeling, you know, confident, and made us feel better. Autumn, she was always around for us, like doing the same, making sure that we were, you know, doing what we needed to do, make sure like everything was like done on time. And she also like gave us rides to like all the different field trips too. And the PEP interns, they've made this experience like really amazing, and it's just really sad that it's over now. These are all the science institutions and my resources. Are there any questions?
Looks like in Falmouth, the last few years have been really rainy, and I wondered if you could tell us anything. Is it like mostly there's been a few big storms, or is there, I know you tried to pick it apart by season. Do you have some idea about like when, like, can you describe that?
So like all this coding, it was like pretty extensive, like data, like it was like some of them were like 20,000 rows. So I did spend a lot of time on the coding, but as far as like the further analysis, I guess that's something I could have added in there, is like look into like those years in particular instead of at like a larger scale to see what was going on. But I do believe there was like the storm intense. That was an intense storm year. I believe it was 2021. I don't know if maybe that was like a hurricane that I'm from Louisiana, so like if we get a lot of hurricanes, that's where my mind went. But yeah.
Did you look at the so Falmouth looked a lot more moderated than like the South Coast cities. Did you look at what our nearby water temperature trends are?
I did not, but there was an article that I didn't mention in this, the Atlantic Meridional Oceanic circulation. It is slowing, which is something that I guess helps move the cooler and water gradient of like the different waters. And it's slowing it's causing the Gulf Stream to increase and like warm up the waters. So I believe there is like a time into that.
So Jordan, do I understand correctly that the precipitation really didn't change, but the temperature did from like the 1960s to now? And you verified that using actual data from Falmouth and the surrounding areas. Do I understand that correctly? That's pretty impressive, getting real data. Put it to use. Thank you.
That Golden Garage and Kayak Albion and Help the Kite Pow in Ackley. And I was working for the Ocean Twilight Zone and doing analysis of the student community ocean. So a little bit about what the Twilight Zone is and why it is in setting. So the OTZ, or the Ocean Twilight Zone, is the region of the ocean that is about 200 meters to 1,000 meters below sea level. With the zone above it being a pelagic zone, which is 0 to 200 meters below sea level. The Twilight Zone is believed to be the location of the largest migration or daily migration on Earth, which is known as the DVM, or the Deal Vertical Migration, which is essentially hundreds of thousands of organisms coming up from the Twilight Zone to the surface zone, and during the night and coming back down as the sun rises. So a bit of its importance. The reason that the Twilight Zone is so important is that it has a very big food web link between the surface zone and the deep sea. The generally big predators as seedlings of swordfish and the sharks come down from the surface zone to the Twilight Zone to feed, and then come back up. And then as things within the Twilight Zone die, they fall to the bottom within the abyssal zone or the deep sea, and then they are then food for the other animals that live down there. It's also increasing importance in recent years with the push for the 30 for 30 plan, which is a big kind of a big goal for people to have 30% of the world's oceans to be protected by 2030, which if you guys don't know is only in 6 years, which is why. And so the reason why the Twilight Zone is so important to that specific plan is that it's believed to have about 80% of the world's ocean's biomass within its waters, which is a very large percentage. And we also don't know much about the Twilight Zone. So if we don't know, can't protect it. So we also don't know what the data of what it makes up. So what things, squid versus fish, and those kind of statistics. So working in summer, my project was a bit too bold that I worked within the Marine Policy Center using data that I've got from GPIF, which was Global Biodiversity Information Facility, which is a database where a bunch of different institutions and organizations can upload and record their observations of different organisms within whatever zone it is. But I was primarily looking at the epipelagic or surface zone and the mesopelagic Twilight Zone, and comparing those two for their biological observations. So I did the video analysis of those kind of observations within the EEZs, or exclusive economic zones. Sorry, switch. But the exclusive economic zones, which are basically the zones around the coasts of different regions. And I was primarily looking at Alaska and Hawaii. And looking between the biodiversity distributions, the geographic distributions of these observations, and the overlap between these observations with the epipelagic zone. And this is a graph that I came up with that a few years are missing is because it's the only the observations recorded for Alaska and Hawaii. So some years might be that there was none for those, but that the large green bars are the epipelagic observations, so surface zone, and then the blue bars are mesopelagic observations for the twilight zone. And as you can see there, within this database, there's been no twilight zone observations recorded since about 2008, which I thought was very significant, as this recent push for twilight zone discovery has only come in the last 5 or 10 years. And then the second part of my project was working in the Redfield lab with, in the Govindarajan lab with working on gut content analysis of swordfish and identifying different squids that they had possibly consumed and seeing the connection of what kind of squids they were eating and whether or not they reside within the twilight zone or in the And this is a DNA sequence, a forward and reverse of DNA sequence that I had found from this gut content. And in the bottom, there is a picture of what it ended up identifying with, which was a swordtail squid, which is a twilight zone squid. So a couple of graphs that's kind of showing the distribution between these two species that were made by my mentor, Hauka Kai Pao, is showing a clear kind of clear distribution or over distribution of observations within the Alaska EEZ, highlighted portions here, versus Hawaii, which is showing a clear lack of or big missing gaps within its geographic areas of observations being reported. And there's a lot of different reasons that this could have been, because this isn't a required thing for our organization, that this is kind of a self-reporting thing, so if people want to do it again. And these, in Alaska, there's a lot of really major fisheries off the coast. So there's a lot more push to put that data widely available, and it's a big, it's a site of a lot of big shipping routes, as opposed to Hawaii, which does not have a lot of major fisheries or big shipping routes that go through its waters. But going further into these points a little bit is showing that within these points that Alaska Kingdom distributions were showing that most of what they were finding within Alaska is within the surface zone or the epipelagic zone, but very little being found in the mesopelagic or being attributed to these different points. As in Hawaii, that it's about roughly 40% of the recorded observations being within the mesopelagic versus so even though there are more recorded observations within Alaska, there are more twilight zone observations recorded in Hawaii comparatively, relatively. And also to kind of show actually how much more information there is in Alaska and Hawaii, there's about 70,000 occurrences shown that were recorded in Alaska, and there's about 4,000 for Hawaii. So that's kind of a really big gap in information. So, and then further breaking it down between epipelagic and mesopelagic between Alaska and Hawaii, that Hawaii, even though obviously having a lot less observation, is showing a much wider range of observations. So what we're seeing is there's a lot more there, as opposed to Alaska, that it's kind of showing just a smaller number of observations, even within the mesopelagic, that there's a lot of the same things being found, or the same kinds of things being found in Alaska versus Hawaii, which is also showing its identity as a big hotspot for biodiversity and kind of confirming that within So talking a little bit about the second part of my research this summer is that I was working with some samples that were collected from a 3-ship cruise from my PhD student at Hui named Kiara Willis. Essentially, they had these big nets and they threw them overboard and they trawled through the waters and they caught small, tiny little organisms like here, and then they caught like medium to smaller big fish, and then they caught big predators like this swordfish. So they caught like swordfish, yellowfin tuna, and those kind of predators that are known to kind of dive a little bit deeper into the water to catch things. So from that, from those kind of organisms that they caught, they were able to extract the stomachs of these animals and to then look at the gut content of these animals to see what they're eating and how much of this diet, how much of their diet was consisted of certain kinds of animals. So specifically why we focused on squid is because squids are notoriously hard to catch in these kinds of nets. They're hard to observe. They're very fast. They kind of break apart if they get caught in the net. So it's very interesting to see these kind of things because even though they are partially dissolved, so they might not be able to visibly see, or able to see it from their DNA that they were present, and kind of see the makeup of their diet. So I worked with identifying these species of squid found within the gut content using DNA amplification, which essentially takes one small part of the DNA found in the stomach and replicates it many times, and then that DNA sequence can then tell us what kind of species that squid is. Kind of the overall point of why this all important is kind of just goes back to conservation. It's very hard to say what we can protect if we don't know what's there, and to put in conservation policies and to make a decision about which areas to protect over another is very hard when these kind of gaps in our information is still very prevalent. And these are just two very small regions of the ocean that we consider as opposed to other areas within the ocean as well. I'd like to say thank you to everybody in the PEP program for the news that out here and open to teaching me about new things and what I see for housing me.
That was really cool. I'm assuming from the DNA, you could only analyze squid or only get the species for squid that were already known.
Yes.
So did you find DNA from things that were sort of in the squid group that were never identified or not necessarily?
So by the time I was handling the samples, they had identified them as they were that but they have been able to find some instances that they're able to say, we know that it's this, but we don't know exactly what this is. Which also warrants more into the fact that we don't know everything that's being eaten or the interactions that it has or whatever. So,
right. Very cool.
Yes.
What was your favorite part of this work?
This project.
About this, yeah.
Honestly, I really like the lab work. It's like being able to find like what the squids were and looking at like the images. So it was cool. Like, it was like a matching game. So that was fun.
Phil Smith, and this summer I had the pleasure of working at Woods Bowl Science Aquarium with my mentor. And my project name is Substrate Dependent Ground Patterns and Squillium pusa antlers on their excavation process. So some common misconceptions about mantis shrimp. They are not actually shrimp. They're stomatopods closely related to crabs and lobsters. And when people typically think of mantis shrimp, they think of this guy right there, the really colorful one. That is the peacock mantis shrimp. But there are many different species and types of mantis shrimp. And one big difference between mantis shrimp are smashers versus spears. So spears, you can see this claw right here called the raptorial claw, and they use this to slash and dig their prey. Meanwhile, smashers, it kind of looks like a boxing glove, and they pretty much punch their prey. And another common misconception is people think they're easy to manage, but they are not. They are actually very aggressive and solitary animals. You put a bigger fish in that thing, with smaller fish. So some background knowledge, they are native to the Chesapeake Bay and they like murky temperate water, but you can see them all the way from Cape Cod to the Gulf of Mexico. So their adult length, they can reach about 20 centimeters. That's pretty much roughly 7 to 8 inches. And they have two types of burrows. They have winter burrows and summer burrows. So their winter burrows are typically deeper and more intricate since it's colder in the winter. Meanwhile, their summer burrows is typically U-shaped with two openings and it can reach 15 through 50 centimeters down with a length about 2 meters. And if you don't know what 2 meters is, that's pretty much the size of Michael Jordan, that's 6'6". So some biology of the shrimp, what they actually use to burrow, they use their raptorial claw and their pleoplugs. So with their raptorial claw, they will actually scoop up the sediment and push it aside to pretty much burrow. And their pleoplugs, they will pretty much kick the sediment out of the way. And I have a few videos. So this video is them using their raptorial claw to pretty much grab the sediment, push it to the side. As you can see, it kind of open it up and dig it for it. Pretty much looks like they're running on a treadmill. So yeah, why is this important to study? So their habitat preferences, where you find them, and pretty much how long they burrow. There are different types of burrows in different substrates and sediments. Also, conservation efforts to ensure their survival in the ocean and captive aquatic environments, making sure that the stress levels are low, their activity is high. And also, they have an ecological role and importance within the environment. They pretty much turn over sediment, bringing up new sediment to the surface, and they also oxygenate the area. So my methods meet turbo and bubbles. So so what I did, I set up two 125-gallon tanks. You can see the inflow, outflow of the tanks. And then each week, I will put 7 inches of a different substrate in the tank. So the first week, I used coral reef sand. The second week, I used crushed coral. And the third week, I used fluvial plant and shrimp stratum. And then the fourth week, I did a combination of all the substrate. And you can see in the picture to the left, right, it's pretty much a finished, complete tank. And you might be wondering why a PVC pipe is in there. Typically in captive aquarium environments, they have a PVC pipe in there. So I wanted to see if that is actually inhibiting their natural behavior of barrel. So some visual representations of each sediment substrate. We have the coral reef sand, the crushed coral, and the fluvial plant and shrimp strata. You can see they're all different colors, sizes, and they're different textures. So, and this is the combination of all the sediment together. After this, I kind of like swirled it around too. It looked like cookies and cream ice cream. And then I created a observation log, and some things I was looking at were the number of visible barrels, the number of entrances. What were they doing in the barrel? Was it visible substrate manipulation in the tank? And if it was any signs of stress within those different substrates. So some results. Unfortunately, it was not what I expected. I expected very intricate barrels, but they did not want to do that, and I realized they are very lazy animals. But the deepest barrel, they kind of made like mini barrels. The deepest barrel was 2.5 inches down, and the location of those barrels were typically either in the corner of the tanks or under the PVC pipe when it was inside the tank. So as you can see, it's kind of divot under the entrances of those PVC pipes. And also, at the middle of each week, I would take out the PVC pipe to see the activity while the PVC pipe was inside of the tank versus when the PVC pipe was outside of the tank. So this is a graph pretty much explaining their activity, their barreling activity while the PVC pipe was available to them. As you can see, only one of the mantis shrimp really barreled in those sediments, so its activity was really high even versus activity at the end of each week while the PVC pipe was outside of the tank. You see an increase with both mantis shrimp actually creating more barrels, except for with the fluval plant and shrimp stratum. So the optimal substrate I found was the crushed coral versus the worst substrate I found out was the fluval plant and shrimp stratum, because it was pretty much little mud balls, and one, it made the tank extremely dirty and it stained everything and made the tank cloudy. And every time they would try to actually barrel, they just hated it. So their stress levels was really high in the fluval plant and shrimp stratum. You could see them swimming more than see them kind of running into the balls. And so some further research we could do with these mantis shrimp is seeing their barreling behavior within different life stages, because I didn't know the age of these mantis shrimp and they typically only live for about 3 years. So I don't know if they was reaching the end of their life and really didn't care about barreling. So maybe that even when you get up from the latter stage of their life. And also you could research different conditions they're in, testing different water rates and temperatures of the tank to see their environment activity. Also, you can see time as well. I only had 7 days for each substrate, so extending the time periods could also be a factor. And then analyzing particle sizes for the different substrates. I would like to thank the PEP program, Andre. I would like to thank the Science Aquarium and Dr. Katie and everybody else that worked with the aquarium, Sarah, Julie, and Allison. And I'd like to thank you all for listening.
Two small ones. First of all, do you think that the size of the tank would matter? Like in a bigger tank that would do more burrowing, maybe? Yeah, go ahead.
I think they would, if they had more space, they would be more curious about their surroundings, because when they did start to burrow, typically it would be like at the ends of the tank. So they realized the tank was right there and it was like, what's the point of burrowing? If they had more space, most likely.
And you said basically these guys have summer cape houses, I guess, and they're different than their winter homes. And you said that the summer ones are like U-shaped. Do you know why? Like maybe for better, I don't know, water flow or something?
I'm not too sure why. I was reading a research paper on it, but it didn't go too deep into why they need those U-shaped barrels. But then the summer one, the winter ones, usually is more openings and it's more intricate, more tunnels. So yeah. Yes.
So I know that you had an option of different projects you could do this summer. What made you pick doing this one?
So that's a funny story. Actually, I go to the University of Miami, and for my college essay, I wrote about the peacock mantis shrimp. So I was like, I'm gonna just keep going, keep working with mantis shrimp.
Hi, my name is Corinne LePage. This summer I worked at Lee on a geochem project with Dr. Lawrence Martin and Dr. Peter Barry. The name of my project is Geochemical Monitoring of Lavas Measuring Helium Isotopes in the 2018 Kilauea Volcanic Eruption. So my project focuses on the big island of Hawaii, the Hawaiian archipelago actually, which is officially named Hawaii. The Hawaiian archipelago was formed over millions of years by hotspot volcanism. What is a hotspot? So a hotspot is an area in the Earth's mantle where a plume rises, forming a volcano on the crust. Over millions of years, the Pacific plate drifting over the Hawaiian hotspot, kind of like a conveyor belt, that formed the island. So Hawaii is presently positioned over the hotspot, fueling a number of volcanoes, including Kilauea, which is highlighted on the map as the red triangle. Volcanic eruptions are one of Earth's most powerful processes, and they can significantly impact the environment and human society. Kilauea has a long history of erupting and causing a lot of damage and hazard. Between May 3rd, 2018 and September 4th, 2018, Kilauea erupted and it caused 716 homes to be destroyed. More than 3,000 people were displaced. There was destruction of natural habitat. There was death and displacement of wild animals, livestock, and pets. 32.3 miles of road was destroyed. 14.5 miles of water lines were destroyed. 900 utility poles were destroyed, so knocking out electricity. And there were huge economic losses. For example, 415 million in tourism revenue was lost. So the damage, displacement, and possible death that volcanic eruptions have the potential to cause is very real. So all of these pictures are from the 2018 eruption. So can you imagine just standing in your house and looking out the door, seeing a wall of lava, like as tall as a tree line, coming towards you? That's horrifying. So what can you do to mitigate these volcanic disasters? So one option is geochemical monitoring of volcanoes. My project this summer looked into enhancing volcanic hazard prediction through geochemical monitoring, specifically through the examination of helium-3 and helium-4 isotopes. So before getting into the exact details of my project, I wanted to take a minute to briefly touch upon helium isotopes. Helium isotopes are noble gases, and they're chemically inert. That means that they can't be chemically altered, so the readings will remain accurate, even in caustic environments. Sorry. Oh, there we go, okay. So helium has only two isotopes, helium-3, which is primordial, which was caused at the beginning, like when the Earth was formed, and helium-4, which is radioactive decay from uranium and thorium. Helium is measured as a ratio, with air being isotopic standard for terrestrial helium at 1. So that means, what is it? that means air equals one. Yeah, so the ratio goes down when there is more helium-4, like in the crust. And when there's more helium-3, the ratio goes up. So you can see there's more in the mantle, and there's even more in like the mantle hotspots. So here's a hotspot, like a plume. There's a lot of helium in the mantle, and it rises up in magma and gas through these hotspots. Remember, Hawaii is a hotspot. So that means long-term eruptions are typical of island hotspots. Long-term eruptions can be useful for geochemical monitoring, and an increase in helium isotopes may provide warnings for hazardous eruptions. The 2014 eruption of Mount Ontake in Japan is an example in which long-term geochemical monitoring might have helped prevent tragedy. Levels of helium-3 and 4 in hot springs increased over 10 years, leading up to an eruption that was not preceded by abnormal seismic activity or ground deformation. So basically there was no earthquakes, there was no bolting in the volcano, so they didn't know it was going to erupt. Unfortunately, the helium increase wasn't connected to the eruption until afterwards. So this graph, this previous study was done, and this partly inspired the research project I'm involved in this summer. So this graph is a decadal time series analysis, and it shows the helium increase. So if you look at this blue bar, you can see that in about the year 2004, there's an uptick in helium that goes all the way until 2014, when there was the eruption. Let's dive into the process of my project and talk about measuring the chemistry of gas. Dissolved volcanic gas is held in magma by pressure. During eruption, the gas is released. This is like when you shake a can of soda, pop the top open, and all the physics blows out at the top. So we obviously can't stand at an eruption site and catch volcanic gas with our hands. So how are we going to measure the gas? Like, how are we going to get it and measure it? One way to measure the gas one way to measure the gas is trapped in rocks, which is what I did for my project. Lava is molten rock, but it can also refer to the hardened rock that forms when molten lava cools. So you get something like this. These cool lavas have minerals in them with which gas is trapped inside. So all this green stuff is minerals. For my project, I extracted olivine out of lavas deposited on different days of the 2018 eruption in order to measure the helium inside. So the lavas were crushed down and pulverized, and I was able to extract the mineral separates of the olivine, which is this stuff. This is actually the olivine I did extract. I picked this under a microscope. I collected a bunch of samples and put them in little glass vials. A fun fact that I wanted to share is that many people are familiar with olivine, you just don't know. So it's actually a mineral of a very popular green gemstone. It's peridot, which is the August birthstone. Since it's August, I thought I'd mention that. So anyways, like I said, I put the minerals in glass vials. Each vial had to be at least 0.5 gram in order to be measured in the mass spectrometer. So what's a mass spectrometer? After picking the olivine separates, I had to run my samples in the mass spec located in the Barry lab at Hui. So like I said, what is a mass spec? Basically, it's a machine, it measures gases. A very brief description of this process is that the olivine minerals are crushed under a vacuum to release the helium and other gases. So they're inserted over here, and they go through this line. So the gases are passed through the extraction line, so it goes through all of these things. And then it eventually goes into the mass spectrometer, which is over here. And that's what measures the gas. So these are my results, which I have 3 points on this graph. These results show helium levels of lavas from different days of the eruption. More specifically, I have, this is day 5. This is day 25. This is day 94. So no significant rise in helium, can be seen on this graph. But the graph is kind of incomplete because there are way more samples that need to be run, which means that there'll be a lot more data for this graph. So results may change. And these are like some more conclusions. So decadal time scales have successfully measured increases in helium leading up to hazardous eruptions. This proves, that it can be measured. Helium increase was not seen like throughout the 2018 eruption yet. So like that doesn't mean that it won't be when more of the samples are run, but it might also just be too short of a time period to actually see like a significant increase in helium. And then so what's next? So I learned some amazing things about geochemistry this summer. I am not a geology or a chemistry major. But I got to explore this area of science that I normally wouldn't have thought to explore without the PEP program. So I'm really grateful for that. Helium is my summer project. So measuring helium is my project, but it's not my passion. I think it's really cool. And I'm encouraging people to do it. But my passion is like environmental conservation in Native American and Indigenous studies, specifically looking into like non-Western systems of science and comparing them to Western systems of science and seeing how they can ethically combine in like in mutually beneficial ways, right? So this map I created to kind of like show my interests and also like build off of like the project that I did this summer. So the red on this map are all the volcanoes. The purple is native homeland. To be clear, all of Hawaii is native homelands, but this definition of native homeland is just government definition, so it's just the areas held in trust for Native Hawaiians by the state of Hawaii, by the government. And the green is extraction sites for this project. So they're the lower Eastern Rift Zone extraction sites, and that's part of Kilauea, that all the samples were taken from, some of them that I worked on this summer. So you can see that in the green. I made this so like best practices basically. I think from now on, anytime I'm involved in any kind of like field work, or sampling, I think it'll be really cool to make this map, especially when you're working with Indigenous peoples and local communities. So you can see like if you're, like on their land, where you are, just, I don't know, because really cool. So that's pretty much it. And let me click over. There you go. So acknowledgements. So Dr. Peter Barry from Hui was my secondary mentor. Thank you so much, especially for helping me put together this project and explaining to me the same thing like 50 times in 50 different ways. Dr. Ben Heiner from PEP, who inspired me to try R. Dr. Forrest Horton from Hui, I wouldn't be here because he's my mentor, like without taking me into the lab, thank you. And having faith in me because like I said, I have no geology or chemistry background. Anji, thank you so much. Like you've been a real one throughout this whole process. And Dylan is from Hui, who I would not have been able to make those bar graphs without basically just everybody in PEP. Like y'all are so good. So thank you.
Just wondering, in addition to helium, did you look at any other gases, argon, for example, or?
So no, I didn't. My project was specifically like on helium because that's what like it's mentioned. But yeah, there's other ways. People, I'm sure there are people that believe like it's different gases. But yeah, just helium for me.
Very cool.
Thanks. that can either prevent or facilitate disease. If there's a bunch of good bacteria in your mouth, then it can prevent disease. And if there's a bunch of unhealthy bacteria in your mouth, then these biofilms can allow for disease. So the structure on the right over here is a hedgehog structure. And this is a structure that's usually found in the mouth in a healthy microbiome. And all these different colors are different types of bacteria. So not all bacteria is bad bacteria, and there's a bunch of it in your mouth, so we should be aware of it. So these bacteria are able to aggregate so closely together because they're exchanging nutrients. And the in a laboratory setting, but again, are not good for the controlled environment that we're trying to create. So along with using this media, we also use a minimal media that has like the base things for bacteria to grow, but not everything. So bacteria need phosphorus, nitrogen, and carbon to grow. And the way that we're trying to get these Streptococcus cells adhesions to work is by giving it phosphorus and nitrogen, but not giving it carbon, and hoping that it would attach to Cranibacterium to try to use its reserves for its carbon. So the way that I would replace rich media with minimal media is by first growing our Streptococcus cells overnight in the rich media, and then spinning it really fast in this machine, and then taking the rich media out and replacing it with the minimal media and incubating it in a now starved setting. And because this was like our first time starving cells, we wanted to make sure that getting rid of the carbon source still allowed for these cells to grow. So what I use is a spot dilution method to get how many colonies formed per concentration of cells that I was making that week. So I would take the big stock that I just incubated, and then I would dilute it, and I feel like the pictures is better at explaining this. So I would put it on a plate and let it evaporate, and then I would dilute it by factors of 100. So from 10 to the -1, it just looks like a huge big like white circle. And I don't know if you can see, but at 10 to the -5, there's like a bunch of small colonies instead, and that makes it easier for me to count. So this was a way to figure out that our cells were alive and they're high in concentration. And then I would just do some calculations, like if I counted 12 colonies, then I would multiply that by how much I diluted it by, and then divide it by the volume of culture and get how many colonies per stock of that day there was. And this is basically like a figure of that. So on the first day, there's like millions of cells, 900 million cells in this stock. And then on the second day, they died down a significant amount, but they're still somewhat alive. But by the third day, there's virtually no cells, which makes sense because we are starving. So now we're gonna get into microscopy and finding out what starving cells actually work. So before we did that, we have to go back to our Cranibacterium because it is the base for our formation. So we tested two things. We tested two things to make sure that Cranibacterium worked as an effective base for adding Streptococcus. So on the left is Cranibacterium on a microscopy slide without any saliva. And then on the right is with saliva. So we wanted to make sure that there was enough surface area for Streptococcus to attach without any problems. And then we also washed the slide once we added Cranibacterium to make sure that that anything that we were adding on top of it was actually attaching and not just in close proximity to it. Just to make sure that our microscopy results were correct and that they looked good. So after we added the Cranibacterium, we then added the star cells on top of it and fluorescently dyed them so that when we put them under the microscope, we can see them in different colors. And we used a confocal microscope to do so. So did starving Streptococcus and Cranny. The starving Streptococcus and then adding it on top of Cranibacterium work to create corn cob structures in it. So a little bit of background before I like get into the pretty pictures. The pink is the Cranibacterium, and inside of some of them you can see like it's stained green, and basically these are just the nucleoids being stained. So all of our cells were stained with the green dye. And then the Cranibacterium was also stained with the pink dye. So for the corn cob formation, I feel like the top left and the top right, the bottom left images are good at showing what a corn cob formation actually looks like. So you have the long pink cells, which is the Cranibacterium, and the small Streptococcus cells surrounding it, which means that corn cob formation did work, and starving the strep cells did help create this type of formation. So some future directions of this project, analyzing gene expression. So we know that starving our Streptococcus cells regulates the adhesion to that Cranibacterium, but we're not exactly sure why. And we also didn't do any other conditions besides starvation or combine starvation with any other conditions. We're not sure what can delay the attachment time or inhibit attachment time or speed up attachment time. We didn't do like too many other factors, so I would like to do that. And then we kind of did a look at Streptococcus and Cranibacterium after it attached. It was kind of a one and done thing. We saw the images that we wanted to see and kind of left the slides alone. And in the 1980s paper, they did some scanning electron microscopy with their slides, which I would have liked to do just to see the attachment closer up. So these white cells would be our Streptococcus cells, and this like grayish line here is the Cranibacterium. And I would have liked to see my cells look like that. So I wasn't too big on the health implications of microbial structures just because I was working more so on the how this happens or just trying to get it to happen in general. But corn cobs are one of many microbial structures in the mouth, and I only looked at one microbial structure in one site. So this model, I guess, can be used in other studies, and understanding bacterial interactions in a healthy microbiome can help when also trying to understand how microbial communities act in unhealthy microbiomes and maybe prevent things like cavities and gum disease. So thank you. I want to thank the Forsyth Institute and the Marine Biological Lab for allowing me to conduct research there, and also for allowing me to be in the lab group. Also the 6 institutions or 7 that held a bunch of panels and let us see everything that was going on behind the scenes. And thank you to my PEP cohort. Love those guys in Anji and Autumn. Yes, any questions?
Well, are these microorganisms only found in human mouths, or is there also in other organisms there?
I'm not sure. Our lab group, they don't see human microbiomes, so I'm not sure. And also these exact cells are not always found. The microbial structures are found, but not like the exact bacteria is found in every human mouth.
And other like bacteria, microorganisms make the corn cob structure, or is it just?
Yeah, so there's other cells that can stick to bacteria for corn cob structures, but most times it's Streptococcus that's found attached to it.
Hello, my name is Amari Johnson. I'm a student at Prince George's County Community College in the state of Maryland. And this summer I worked on a salt marsh restoration project with Seth Ackerman and Megan Eagle at USGS, where my focus on this larger project was in remote sensing and the hydrologic processes of an engineered salt marsh, with hydrologic processes being the water flow in and out of the marsh. So what is a salt marsh? Salt marsh is essentially a swamp mixed with a grassland. They are normally located within estuaries like the Chesapeake Bay. And in fact, the picture on the top right was taken by the Chesapeake Bay Foundation. There are 2 types of elevation marshes or marshes. There's a high elevation salt marsh and a low elevation salt marsh, and this depends on how often areas of the marsh flood. They also provide habitat for animals like the salt marsh sparrow, which are critically endangered due to rising sea levels causing the marshes to disappear, which causes the nest of the salt marsh sparrow to flood. Yeah. So in order for a marsh restoration project to be successful, it needs these components. It needs to have hydrous soil, or soil that is so saturated with water that it creates a low oxygen environment, which prevents bacteria from decomposing all dead material quickly, which allows the plants to take up more nutrients. It also needs reference sites, which are going to be local marshes, natural marshes to the area, as well as any other successful marsh restoration projects. As mentioned earlier, it should be resistant to erosion, as that is a major issue with salt marshes, and it needs to have the right vegetation. Vegetation is extremely important to a salt marsh restoration project, as their roots provide structure and they also filter out any harmful toxins that may flow into the stream from the groundwater. They provide shelter to animals, like mentioned earlier, the salt marsh sparrow. And they also add elevation and flooding, as well as vegetation, plays a huge role, correlates to each other. So in the upper marsh regions of a salt marsh are going to be the trees and shrubbery, with the middle marsh containing most of the plants you would typically see, like in this section of the photo. And the pioneer zone, which is going to be near the water's edge, is going to be where you experience plants whose seeds flowed into the marsh from an outside source, with any plants below that being seagrass and microalgae. So HAT, or HAT, stands for highest astronomical tide, which is the highest the tide has ever gotten in a 40-year period. Mean high water springs is how high the tide normally gets within a 24-hour period, with mean high water neap being how low the water normally gets within a 24-hour period, and mean low water being how low the water ever gets, which is normally going to be during a drought. So this brings me to the marsh I'm specifically worked in this summer, which is called Marsh Island. It's located in New Bedford Harbor, which is in Fairhaven, Massachusetts. It was destroyed in 1871 when it was converted into a granite quarry. And restoration began in 2023, with this specific marsh being engineered to be a high elevation salt marsh, which allows it to be more resistant to erosion, although that it's not immune to it, which I'll show in a later picture. Phase 1 of this project is complete, which is this upper section of the marsh, with the blue crevices going into the marsh representing the stream, and the yellow line going through the outskirts of the marsh trail that would allow the public to walk through. And they also built a bridge at the entrance of the marsh. This is a picture of the marsh, which was taken from the bridge mentioned earlier, where we can see some of the pioneer plants, younger pioneer plants experiencing flood events. This is a picture showing erosion happening at the marsh, which is in this muddy area, which shows that even just a year after being completed, Marsh Island is already experiencing significant erosion. So the remote sensing part of this project, my methods for the goals for the remote sensing part of my project was to obtain Marsh Island's elevation. I also wanted to calculate how the areas of the marsh that flood and for how long those areas stay flooded by a percentage, as well as how flooding might affect the vegetation in Marsh Island. So for the remote sensing part of my project, I used a drone with a normal high resolution camera to take a picture of the entire marsh. I then imported those pictures to a software called Metashape, where I created a digital surface model and an orthomosaic, which is essentially an aerial image stitched together by those pictures. I then extracted the drone's data set from that orthomosaic to use in R for my hydrologic analysis. This is an oversimplified version of what I did, but I essentially broke the marsh into 36,000 grid cells, which represent the areas of the marsh. And then I took the extracted marsh elevation, combined that with some water elevation to see which areas of the marsh were flooding and for how much water was in each grid cell. I then used that water volume in each grid cell to calculate the percent time flooded. This is a digital surface model of the marsh, showing how high each of the areas are. The red represents 10 and up in elevation in meters, which is going to be the trees and shrubbery that was mentioned in the upper marsh regions. The blue is going to be -3 in meters, which is going to represent the stream as well as New Bedford Harbor surrounding the marsh. And the trail mentioned earlier, which makes up the border of my study area, is around 4 meters in height. So the highest elevation within my study area is going to be 4 meters. And the area within that border is my study area, which is around 1 meter in height. So this is a graph showing the water elevation in meters. So the x-axis is the date in months, with the sampling period being from December 2023 to July 2024, with the y-axis being water elevation in meters. So each point on the graph represents a time in that day that the water elevation was sampled, and it was sampled in 15-minute intervals, which is why there are thousands on this graph. But the most noticeable points are the ones near the 1 meter mark, which there are very few of. And as mentioned earlier, most of the marsh doesn't go above goes above 1 meter. Which means that most of Marsh Island is not flooding that often. So this is a map showing which areas of the marsh are flooding and for how long those areas are staying flooded by percentage. With dark red, which is the trail mentioned earlier, representing 0 to 1% of the time, with the blue being the stream flowing into the marsh, which is going to be flooding 25 to 100% of the time. And the inland part of the marsh, which is where I mostly obtain my marsh elevation data, floods around 3 to 4% of the time, as well as some greenery within the borders of the stream, which flood 10 to 25% of the time, which is represented by the green. In the next slide, I'm going to zoom in on this little area of the marsh to show the erosion as well as why some of these areas are dark red. So where the arrows are pointing is where some erosion is occurring, which is going to be these dips into the marshland itself, as well as over here in this picture. And the dark reds, which once again is represents flooding 1% of the time, is not correct. But the reason why it's showing that is because there's dense vegetation right there. So the drone had a hard time seeing through that vegetation. So we couldn't get accurate marsh elevation data. But this can also help infer that these plants are thriving with the current elevation, as they're vibrant green and there's an abundance of them in this area. Even the plants in the upper marsh region, which only floods 3 to 4% of the time, are appearing to be extremely healthy and thriving, as there are patches of them growing, and they almost appear as blades of grass. grass, which means that the current elevation of Marsh Island is almost perfect for the vegetation to grow within their respective areas. Another aspect of hydrology that I focused on was the water flux, which is the flow of water in and out of the marsh, but I won't go into that in today's presentation to save on time. But I just wanted to mention that this was another part of my project I worked on this summer. So what can my results be used for? They can be used for documentation or making sure how the marsh is doing and its management. It could also be used for vegetation management, I mentioned earlier, to make sure the health of the plants is being maintained. And it can also be used as reference for any future projects, salt marsh restoration projects that USGS or any other organization may conduct. I just wanted to thank Median Eagle, Seth Ackerman, Anji, Ben, Autumn, my PEP cohort, the PEP funders, SEA, USGS, my family, and Professor Cyrus at my school for giving me this opportunity and supporting me throughout the way. Any questions?
How do you change the elevation of a marsh? Like you were saying, the marsh island, it was being restored to be a high elevation one. Like, how do you do that?
You would essentially add more sediment to the ground, more soil to the ground, and try and even it out so that you can like strategically control that elevation.
Do you need to keep supplementing the soil or sediment over time due to like erosion or weathering, or do you just do it once and you kind of see what happens from there?
I think for now we're seeing what happens as there is erosion around the areas, and it seems like they are trying to maintain that or manage that, but it doesn't seem like they're placing any more sediment right now. And it's probably because this project is early in its development, so they're trying to see if this is really worth all the money and time. Yeah.
I'm just wondering if you know what plant species, what grasses were planted? Were these aquatic species, terrestrial species?
So the plants that are planted are the ones that are in this kind of grid area.
Yeah.
Away from the stream. And those would be more terrestrial kind of. They would be, which is why they're more inland and they don't require as much flooding as the pioneer plants, which are plants whose seeds came in from an outside source. And then they kind of just rooted themselves on the banks of the marsh. So those will be more kind of aquatic plants.
We have a question from the chat. How much of the marsh area does the restoration project hope to flood?
Floods. I'm not sure about flooding. But I do know it's trying to restore 11 acres of the marsh right now. So half of it being restored is probably 6 or 5 acres.
I just wanted to say, I'm glad that both our marshes and our dunes kind of coincide with one another. I know there aren't too many dunes over here in this location, but I was curious, like, are there other marsh projects that are happening around this area that you know of?
Yes, that I know of. I really only know of Marsh Island, but I do know that around this location, a lot of marshland has been destroyed. So most likely in the future, there will be more projects to restore those areas as well.
For your spatial analyses, did you do all of that in R, or did you use like ArcGIS or anything like that?
So to create the DSMs and ortho mosaics, I use QGIS. But to calculate the flood percentages and things like that, I used RStudio.
They're beautiful maps, that was just.
Hi, everyone. My name is Emmanuel Bogomoli, and this summer. Where exactly do you guys feel what I'm doing here? swap the other screen. Okay, so I worked with NOAA Northeast Fisheries Science Center on a project about fin whales. It's titled Small Home for a Big Whale, Fin Whale Use of Marine Protected Areas. And my mentors were Sean Hayes and Danielle Chelua. So first, what is a fin whale? So fin whales are the second largest whale species as well as second largest mammal in the world. They're protected under the Endangered Species Act as well as the Marine Mammal Protection Act. And so off of their physiology, they're very sleek, they are streamlined, they have a V-shaped head, they swim extremely fast, and they are gulpers. So they, take big gulps of food and can eat up to 2 tons of food a day. So they have a very high risk of vessel strikes and fishing gear entanglement, and because they're found on deep offshore waters, there's really not a lot known about them. So why does this matter? Why should we care about fin whales? Fin whales, as well as just whales in general, are extremely important in ecosystems because of the large amount of carbon that they store because of such a big animal that it is. When they sink, as you can see in this picture over here, when they sink, something called a whale fall, and all of the carbon that they have in their body gets trapped for centuries, thousands of years. So that also can create new habitats and feed multiple organisms on different trophic levels for a long time. As you can see here, they're feeding on that whale fall that seemed to fall a long time ago. Additionally, there's something called the whale pump, which is demonstrated here. So the whale pump is when, because of the diving behavior of whales, they bring deep sea nutrients from the deep sea up to the higher in the sea, and that fuels CO2 dependent phytoplankton. And then additionally, when they migrate from nutrition high feeding grounds to nutrition poor breeding grounds, they're transporting nutrients, which is also triggering the phytoplankton as well. And then this graphic shows how actually fin whales have the largest mammalian biomass out of both water and land animals. So because of this and all the carbon that they sequestered, they have very large recovery potential and can help. There's been data that shows that this can really help with healing our ecosystems and their health. So first, to start off, what is a marine protected area? A marine protected area defined by NOAA is a defined region designated and managed for the long-term conservation of marine resources, ecosystem services, or cultural heritage. There's almost 1,000 marine protected areas in the United States, and their purpose, again, is just to protect the marine ecosystems, preserve biodiversity, and maintain the resources for the future. And then there's different types of marine protected areas. There's variety in them. So some are no take zones, which means there's absolutely no extraction allowed. There's no fishing allowed, nothing like that. And 3% of US waters are in no take zone. And then there's other types of marine protected areas that allow things such as commercial fishing, recreational fishing, research. And then there's something called a National Marine Sanctuary, which is a type of marine protected area that is designated by NOAA or Congress. So we'll be focusing on Massachusetts for this. So Massachusetts has 5 marine protected areas. As you can see here, they are mapped out on this. And then we also have the only National Marine Sanctuary in all of New England, which is the Stellwagen Bank National Marine Sanctuary. Stellwagen Bank is pretty important because it actually is home to a couple of shipwrecks, so that makes it historically important. It is one of the marine protected areas that does allow both recreational, commercial fishing, and a lot of whale watching. So when we went out on whale watching in Provincetown, that is most likely around where we were. And then, yeah, so that is the marine protected areas. So on to my research questions. My research questions were, what are fin whales' usage of local marine protected areas? What is the site fidelity of the animals tagged, and how does it vary by individual? What are their behavior patterns, and how is their diving characterized? So how did I research this? My methods. We used LIMPET tags, which are low impact minimally percutaneous electronic transmitter tagging. These are satellite tags that are deployed with an air rifle that puts 2 barbs in the fin whale's dorsal fin. As you can see over here, this is right when it's about to get tagged or already has been tagged. And then this is actually one of the tags It's just a small little tag, but it provides so much data. These tags, you get to actually program them beforehand to measure various variables such as dive depth, dive location, dive shape, things like that. And then lastly, using all these tag data that I was given from both 2022 and 2023, I used R, RStudio, and GIS, which is all statistical analysis and mapping, to come to my conclusions. So also one other thing I wanted to add was that this summer I had a really great opportunity to go into the field in Eastport, Maine. Basically up there, their attempt was to they actually were able to get 2 tags on so far, I believe, and they're trying to tag whales up in the Bay of Fundy, fin whales, to see whether, you know, there's a difference between those whales and the whales that we have data from down here. So being able to see how the, you know, the project is set up, being able to go out scouting and, you know, going on the top of the boat, all of that stuff was really awesome. I got to see how the tags are programmed, and it was just really great to see all of the background to my actual research. So this was a really great opportunity. So now on to my findings. So first, tag, I want to give background on the tags. So we had 13 tags overall. There were 7 from 2022 and 6 from 2023. As you can see, there's a really large variety of how long these tags lasted, and that really, we don't really know why or how some lasted longer than others. But so the shortest one that ever lasted was 1 day, and then the longest was 92 days. So, you know, this sets us up with very different stories of the whales' lives, and this is just one. I mean, even just 92 days is a very small snapshot of how long these whales really live. So first, diving behavior is shape. So, so first I just want to explain. So a square dive shape, that is kind of when the whale goes down, stays down like in the same area for a bit and then goes back up. And those were the most common ones. The ones, square dives that went below 5 meters were 60.7% of the dives that we recorded. Then there's square dives that went above 5 meters, which I'll explain in a second. That was the second largest at 19%. Then there's U dives, which is, you know, again, when they go down and then up, or in like U shape. And then V dives were the least, which is just down and straight up. And we believe that the square dives are foraging behaviors, whereas the V dives are more traveling behaviors. One really interesting thing that we looked at though is these dives under 5 meters. We were really confused because these are such large whales, how can they be diving under 5 meters? And what we discovered is what we suspect after I put this on a dive count by hour and dive type. As you can see here, the squares under 5 meter dives mostly happen in the early times of the day. So what we suspect is that this might just be a resting behavior. You know, the fin whales are resting, staying still, going down and up. Because they are such large whales, that even just a bit of down and up motion could trigger it to count it as a square dive. So this was pretty interesting and something to explore further. So then the next thing that we explored was depth. So this is the frequency distribution of maximum depth by dive shape. As you can see, most of the most that they dived around was, you know, like 15 to like 25 meters, which lines up with the average that I found, which was without counting the dive shapes under 5 meters, the average was the 20.25 meters. So that, you know, that lines up. And the deepest dive that they had was 66.5 meters. So there really is a lot of variability again, and these animals can dive very deep. Next was their dive duration. So the longest non-outlier dive, because we did have some that we believed were kind of outliers because whales cannot hold their breath for that long, was around 10.73 minutes, which is, again, is like shown here. These, as you can see, the square dives were probably the longest ones, the most abundant ones, and it was more abundant between like 4 and 12. And as you can see here, this was where we suspected were like the shorter dives that were shallower. So then one thing that was really interesting that I got to look at was the mapping usage of the marine protected areas. So I made this graph, which there's a lot going on here, but the red is the marine protected areas, and then the black dots is every single location data that we had, and the blue ones are the ones that are within the marine protected areas. So that differentiates it. I also did it by individual. So as you can see, the individuality did vary quite a bit, because down here, this orange one is the only one that really went down here, whereas this green one over here was starting to leave. Sadly, that tag fell off, so we didn't really get to see where it went. But so this was pretty interesting to look at the different individuals and how they differed. And one thing I want to add too is this is a diagram of the shipping lanes, this big gray thing here, the shipping lanes off Cape Cod, and as you can see, it seems to intersect quite a lot with the locations that they are. One thing that I did not get time to do is to, see how the vessel strike threat is increased or decreased based on their behaviors. So that's something to further look into, but I just wanted to show that. And then another really interesting, so aside from just color coding all the different points, I also split them up into separate graphs for every individual. And this is just really like some three that were super different. Like this one really just stayed up here, whereas this one traveled all over. And again, this one was starting to leave. And we even had like one tag that in the top that we had a tag never actually went into a protected area. Because again, this is just one snapshot of such a long life of these whales. But this was very interesting to see as well. So, so one of the biggest takeaways that I found was that the proportion of the points that we had of all the tag data that we had, 37.94% of them were found inside of a marine protected area. So this is a pretty high number, and it means that they are using these marine protected areas at a disproportionate rate. So this moves on to conclusions and discussions. What can we get from this? First, marine protected area usage. Now that we found that they used around in the tag data that we had, around 38% of the points were in a marine protected area. That means that we can now use this to see how we can better protect and strategize to help these fin whales. Second of all, it just shows us that there's still so much to learn, because most research in the Northeast Fisheries Science Center is focused with northern right whales because they are so endangered. There's just not a lot known about fin whales, and there's just so much more to explore. The diving types that we were able to see was really interesting, especially with the one that we suspect is a sleeping behavior. I definitely would like to see that studied further. And then what does this all tell us? So what does this all tell us about, how are these animals using marine protected areas? How can we further help them in their conservation because they are endangered? And, just how does their behavior also help us understand more about them? And also one really cool point I want to show is these are some currently tagged fin whales off of Cape Cod as well. And it's just, you can see the mapping like in real life if you look it up on our tagging data, which is super interesting just to see all around where these whales go. And even here, they're also having extreme individuality. So lastly, here's my references. And then lastly, my acknowledgements. So I'd like to thank my mentors, Sean and Danny, and then members who were in the field house with me, who got me up there, who got me on the boat and made sure I was safe. I'd like to thank Alicia and Laura from Northeast Fisheries Science Center because they helped me a lot with my coding. And then just in general, PEP cohort, I'd like to thank Anji, Autumn, Ben, and Andre. And just here's some cute photos. So yeah. Slide, but does anyone have any questions?
So now that you know the frequency and usage of marine protected areas, where off the coast of New England would you propose a new marine protected area from the fin whale data that you've gotten?
So one thing was, we weren't necessarily looking to like implement a new marine protected area, more just to see, are these marine protected areas working? Like, are they actually using them? And I think because we found that they are using them, I think it's more focusing on how to continue to strategize those limitations on how fast boats can go in certain areas, things like that. But so yeah, I wasn't really looking at like where to put a new one, more of like how much, because we really did not know at all before this, like how much they were using the marine protected areas. And now that we know it's a lot, we're like, okay, so we need to continue to focus on these areas and make sure that they are strongly preserved for these animals. So yeah. Another question. Yes.
So when you went out on the field, were you actually able to tag any fin whales or did you get to see them?
So when I was out in the field, unfortunately, there was some permitting stuff, and so we weren't able to tag them. But on the last day, which was the picture of the boat, we got to go out on the scouting mission, and I really was just there for the vibes, like for the journey. I was like not necessarily looking to see something super cool happen. But around like into our trip, we saw some fin whales, and we just sat there for like 15 minutes watching the fin whales, and that was genuinely like one of the coolest moments of my life, especially seeing them do all of this behavior that I saw on like R and like the Google Sheets data and stuff, seeing that in real life was just amazing, and fin wheels are just gorgeous, like you hear their glow before you see it, like it's just, we saw 3 of them and it was really awesome, so yeah. Yes.
With the way males migrate, move around, do you think that whales act as a nutrient provider or nutrient administrator?
So because the whale pump, whales are definitely nutrient providers, they recycle a lot of nutrients, like I said, the areas where they breed are the areas where they feed are like high in nutrients, whereas then the areas where they breed are low in nutrients, so when they travel, they bring those nutrients with them, as well as when they're diving from the bottom, they're bringing a lot of nutrients up, and all of this is helping with phytoplankton blooms. And there was a bunch of papers that I read that showed that, you know, the conservation of whales can really help with the ecosystem of, in general, marine health because of all the carbon that they bring and the nutrients that they bring. So yeah.
Hello, everybody. My name is Aaron Whitware. I study marine science at Cal State Monterey Bay. And this summer, I'm working with Dr. Andrew Reed and Dr. Stace Boyu at the Local Oceanographic Institute, validating wind data from high latitude. So a little bit about the NOAA. The Ocean Currents Initiative is an NSF long-term project aimed to collect and retrieve quality data. And they do this throughout multiple oceans with multiple different deployments using over 900 different types of instruments. For my project specifically, I'm focused on the Erminger Sea, which is in the North Atlantic, off the southern tip of Greenland. Here's some images of some of the weather conditions in the Erminger Sea. So starting my project, I started off with a background test report of the bulk meteorological anemometer test. And what I focused on was this image here. And this image is showing scalar versus vector winds of our bulk meteorological packages, or MEPBK for short. And what we really want to focus on here is these graphs. These are considered the good data that we have retrieving these different, or looking at this graphic. And looking at this, we have earlier deployments and later deployments, and these graphs are what we consider bad data. I like to call those shrimp graphs because they kind of look like shrimp. So going through this, we really wanted to ask the question, what happened between the earlier and more recent deployments? And while going through this, something that we suspect is that there was a change in software and hardware through the different deployments. So getting into my project, I started an analysis of the scalar and vector components using OOI's Jupyter Hub. And we used this organization sheet here, Google Sheets. And basically, I was going through all of the different deployments that use the MEPBKs. And what I was really aiming to do was to recreate or find new graphs that would support our findings. So these are the graphs that I ended up making compared to the graphs of the Southern Ocean that were previously found in the report. And as you can see here, we have some similar graphs, and then here we have some other graphs that look different. So this kind of supports that finding that we had. More additional graphs, shrimp graph. Okay, so that led into the conclusion of that was kind of like, that's still an ongoing project that we're still trying to find a solution to, but our conclusion was that these instruments had a change in hardware and software. So that led into my bigger project. So during this time, the Erminger Sea cruise was going out to do another deployment. But while I was waiting for them, I had that previous portion. And then when they finally got to their current, their desired location, we started a wind sensor analysis. So the wind sensor analysis was composed of an analysis of these available data sets. So the MEPBK1, both deployments used a 2 GIL axis wind sensor. GIL axis wind sensor. And our MEPBK2 used an RM Young wind sensor. Our direct flux covariant used a GIL R3 3-axis wind sensor. And our 2 ship sensors, port side and starboard, were the Visalia wind sensor. And these were the available data sets that I was able to retrieve from the deployment in real time. And doing this, we really wanted to focus on this specific time frame. During this time frame was when all of the sensors and instruments were actually in the water. So this is when we were able to actually do this comparison and when it was best. So this kind of developed into a little hypothesis of one type of the multiple sensors. We suspect the GIL 2-axis wind sensor has been under recording wind speeds at higher magnitudes. So doing this, I went through a workflow that kind of comprised of multiple different parts. Retrieving the data, which I had previously mentioned right there. So going through and getting the available real-time data set. And then calculating the wind speed from those data sets. So when we're retrieving the data, they don't come to us directly in wind speed or magnitude of wind speed. So I had to calculate the magnitude of wind speed. And I did that using this equation where U is the magnitude of wind speed. And then the U and V are our eastward and northward wind velocity components. And I did that using Python through Jupyter Notebook. So the next part was adjusting the ship sensor to buoy height. So the ship sensor sits on a mast on the research vessel Armstrong, 17.9 meters above the sea level, whereas the buoy sensor sits on a bulk package 5.85 meters above sea level. So, or above the water line, sorry. And as you can kind of see, you can see people over here on like the little ship deck compared to here, very large. So we wanted to adjust the height of this so that we can get accurate readings of the wind data. And that was using the core 3.5 algorithm, an algorithm created by data scientists and oceanographers in tandem groups. So from then I interpolated and resampled the data. So I sliced it to our desired time frame again, that midnight midnight June 10th through 7 a.m. June 11th. When all of our sensors and instruments were in the water. And then I did an interpolation and a resample to 1 minute. So here's a few of the graphs that I ended up making through this process. So this is a graph of the raw data that I was receiving from the ship. We can see that right here is when the ship was deployed. And then through here is when they were when they were traveling to the Irminger Sea. And then about right here is when they ended up getting to the Irminger Sea and when we started our analysis. And this is comparing the 2 ship sensors, port side and starboard side. So then after the time, after the resample interpolation and time slicing, I compared the 2 buoy data or the 2 MEPBK buoy sensors. And I really was just looking to see if there was a difference between the 2 sensors between the new firmware and old firmware. My next graph was a comparison between all of the buoy data, again, the buoy sensors. And this is kind of where you can start to see where our hypothesis is starting to like come true a little bit. Next, I wanted to compare the ship sensors again, just to kind of get another analysis to see what was going on. And then finally, I have a nice, very vibrant plot comparing all of our sensors together, both the shipboard data and the buoy data. In doing this, you can kind of tell a little bit that the DL2 axis wind sensor is under recording a little bit at higher magnitudes, but it's still very, very, very busy, and you can't really tell too much. Kind of looks like a little noodle break. Finally, I did another resampling to 1 hour and averaged everything. So you have this plot, which is the same as that old other one that I had just shown, except you can see it a lot clearer. So I want to focus the attention specifically right here, where we can see that at higher wind speeds, this is our GIL 2-axis wind sensor. And we can see here that it is under recorded compared to all of our other sensors, which proves our hypothesis correct. And in doing this, this is a really nice comparison because we can compare everything that's going on between all of our wind sensors. So our GIL 3, GIL 2, and ship sensors, those are all sonic wind sensors. So we can also test the accuracy of our sonic wind sensors along or compared to our RM Young, which is like a turbine sensor. And I also would encourage everyone to get out their phones and scan this QR code. This is a QR code that goes directly to my GitHub, where I was able to run this notebook. So this is sorry. Just focusing on the GIL 2 axis. So this is a sensor that we use on almost all of our buoys. So it's really interesting analysis to be able to compare this and see why this might be happening. Yeah. So moving forward, something next that we would be doing is making an individual comparison. I started this part of my project, but having more time, I would go through this analysis a little bit more in depth. And we really aim to do this correction or this comparison because we want to be able to develop a correction for what's going on for our history. Here are my references and acknowledgements. I'd like to thank the OOI, and I would like to express my gratitude to Onji, the PEP staff, and our mentors for making this experience memorable and educational. I've gained a great deal of knowledge and insight from it.
If you had the chance, knowing what the urban state looks like, would you go out to work on collecting this data yourself?
Absolutely.
I found it surprising that that sensor so badly failed at what are not super high wind speeds. Is it rated for low? I mean, some wind devices are actually designed for very low winds. I mean, do you know anything about this sensor, why it might be failing?
I personally do not fully understand. That's still an ongoing project that the OOI is doing right now. And I think doing this, doing an individual comparison, trying to make that correction could also help us find out why maybe that might be happening.
So hello, my name is Sinclair Strong. I attended this University National University. This summer, my research was focused on moisture as a driver of net soil methane flux in a northern forest. My research was supervised by Kathleen Savage at the Woodwell Climate Research Center. Okay, so as you all know, all heard of, there's something happening called global warming. It's due to greenhouse gases that are basically accumulating in the atmosphere. Most notably, there are carbon dioxide, methane, and nitrous oxide. So they're trapping this heat in our environment. Usually that goes well, it sustains life on Earth. But because there are there's more of these gases being produced by humans, essentially. The levels are rising and the heat is as well, which essentially causes climate change, as I'm sure you've also heard of. So my research specifically is focused on methane. Methane's incredibly potent as a greenhouse gas. It traps 34 times more heat than carbon dioxide. It's less abundant than carbon dioxide. But it still has like a very sizable impact to climate change. So for forests, there are things called net methane sinks, which are forests that take up more methane than they produce, and then there are also forests that are net methane sources, which release more methane than they consume. Dry forest soils specifically are the largest biotic sink for methane. Research tends to focus on methane sources, so there's not as much done on methane sinks like the one in my research. So this right here, oh wow, this is really interesting. Okay, yeah, this right here is a diagram of the transfer of methane in a forest. Specifically kind of like the one I was researching. So you'll see over here that there are wetlands which are saturated. They're anaerobic, meaning that they do not have as much oxygen. And you'll see that there is a population of microorganisms called methanogens. And methanogens, as you can see by this arrow, this is the flow of methane. They produce methane. And they prefer wetlands because of the anaerobic environment. On the other side, you'll see that there's a population of microorganisms called methanotrophs, which will consume methane in smaller amounts. And they like the upland because it's an anaerobic environment. So right in the middle, kind of, not always in the middle, but you'll see areas that are called transitional soils, which will either produce or uptake methane depending on the conditions. It might be because of rainwater, it might be because of seasons, but there are these three different areas across the drainage gradient. So my big question for my research was trying to figure out at what soil moisture content across these drainage gradients and in a soil profile do fluxes transition from being sinks to sources or the other way around. So this is where my research was conducted. It's the Howland Research Forest. Up here is where it is in Maine, and down here is where we are. Basically, it is a subboreal forest in the northeastern region. There's a lot of research done there because it's a net methane sink, which is a little bit unique. And as it becomes more and more wet because of climate change, increased rainfall, higher temperatures, it might transition from net methane sink to a source. There's also a large variation across a drainage gradient. So as I showed earlier, upland, transitional, wetland, it has all of those different types of areas. And then over here, you'll see me, that's me. And that was me going on a trip to Maine in order to track the methane fluxes at different drainage gradients across the forest. Okay, so here's some of the data that was collected basically from each drainage gradient area. So you'll see that transitional soils, there are a lot of fluxes that are below zero and a lot that are above. So basically what that means is that methane fluxes below zero or negative fluxes would be an uptake of methane, whereas fluxes above zero, positive fluxes would be a production of methane. So you'll see transitional, it has a little bit of both. Upland over here, it's mostly in the sink, so it is consuming a lot of methane. And over here, you'll notice on a completely different scale than over here, because this is like 0 to like 0.05. This is 0 to 1. It is in the production and it's on a very large scale. So the wetland is very effective at producing that methane because it's so saturated. So we collected soil cores that essentially look like this. They're about 30 centimeters long. We collected 12 of them, 6 of them from the upland and 6 from transitional sites. And then we further separated them. So this would be the organic, all the things you're seeing that are brown. So we separated it into an organic top, which was like the first 5 centimeters, so maybe this much. And then we took the rest, and that's the organic bottom. And then we also got the AE layer, which is the mineral layer, the mineral horizon of the core. And so, whoa, don't do that. Okay, so here are the samples. So you'll see each layer like I just described. You'll see the organic top, you'll see the organic bottom, which is the remaining amount. And then this is the AE or mineral layer, which you'll see is paler. So these were the transitional soils and these were the upland soils. Each like row here, this way is a core. And then there are 2 controls, one with water, well, yeah, one with water and then one with air. At first I can't really tell. So my methods for processing these samples were performing 6 laboratory incubations. So in order to prepare these incubations, I wet each sample up. That was by adding 10 milliliters of DI water to each sample and then letting it saturate, letting it sit a little bit, making sure everything got all nice and moist. And then I did an incubation with the light core infrared gas analyzer that basically measured the amounts of carbon dioxide, methane, and water vapor being released by each sample. And then I did some data analysis where basically I calculated the methane flux, which was the change in jar headspace concentration over time. So the headspace concentration is basically the amount of each of those gases in the headspace being the remaining area in the jar if you subtracted the sample. And then I also calculated the percent soil moisture using the gravimetric moisture content, which is essentially the weight of the water divided by the weight of the oven-dried soil. So now we get to our results. So it's kind of a lot, it's kind of little, but it's kind of a lot. So over here's our upland, over here's our transitional. You'll see that this is kind of the range of soil moisture percent, so from 0 to 10 to 20% moisture. Over in the upland, you'll see a general trend where it increases as the moisture increases, the flux increases. So it starts in the sink area and then passes 0 to go into net production, even though it's kind of small. This was the top layer, the organic top layer. This was the organic bottom layers on average. It kind of hovered around 0. It didn't have any general direction for the most part. And this is also on a pretty small scale. Over here for the transitional, you'll see that it looks a little different and you'll find that it starts around 0, so at lower moistures, it was on average around 0. It might have been producing, it might have been consuming, but as you add more and more moisture, that trend is a lot more clear in that it increases as moisture increases in the top layer. And the same goes for the bottom layer, though you'll find that it actually was consuming soil earlier on, and then it started producing as it got more and more moist. So this is for the AE layer. There's a lot less activity in the AE layer. So this, you'll see it kind of hovers around 0, but you'll see like a slight increase. In the transitional, not quite sure what was going on here, but it was incredibly low. It was consuming a lot of methane, but then as it was wet up, it did start producing a little more methane. But of course, if you look at the numbers here on the scale, it is very, very small compared to all other layers. This is kind of the same data, just visualized a little differently. So you can see the clear upward trend as moisture increases. You'll find that the methane flux also increases. You'll notice that in the upland layers, around 30% is where that change happens from being a methane sink to a source. And in the transitional, it's around 30 here, around 40 here, a little lower than transitional, but there's less variation in that, or there's more variation in that, you'll see that that's where the transition kind of is. So for the conclusion, the main takeaways were that increased moisture resulted in an increased methane flux. So transitional soils ended up showing a positive flux, whereas upland soils didn't show as much as a positive flux, but they did come just above 0 in terms of flux, so low levels of production. The transitional drainage gradient areas were much more responsive than the upland areas. And the same goes for the organic bottom horizon, was more responsive than the organic top or the AD layer. We hypothesized that this is most likely due to a more dynamic microbiome occurring in the transitional drainage areas and the organic bottom horizons. Essentially, they are more used to accommodating for drier conditions and wetter conditions, so their microbiomes have developed both populations so that they can thrive either way. So for further work, we need to study microbial populations and basically what their environments look like. So I measure the soil pH a little bit, but going forward, it would be good to measure the soil carbon and nitrogen. And then here are my acknowledgements. Thank you to Marley and Valeria at Woodwell Climate Research, at the Woodwell Climate Research Center for helping train me and also conduct a lot of the things and incubations that I was doing in the field and in the lab. And thank you to these people at PEP for kind of facilitating the program for us. Yeah, that's all. Any questions or this is not down? Cool. Oh, Mia.
We measure like,
For our focus, our scope of the project, we didn't look at the specific microbes in the soil as much, but there's other research to be done on that. That will be done and is being done, but that's good. Okay.
Good afternoon. Welcome to the 2024 Partner Education Partner. Thank you for coming to Symposium. My name is Alex Brown, and this summer I got to work at Marine Biological Laboratory with my mentor, Dr. Kenneth Foreman, and his research assistant, Emma Spathos. And they helped me produce my project, The Importance of Macro Tidal Flux and an Exchange Between Little Pond and Vineyard Sound. So first thing we're going to talk about some background information on Little Pond. So where is Little Pond located? Fortunately, it's here in Falmouth, Massachusetts. So one of the things about Little Pond is it has impaired water quality from septic systems. Little Pond is one of the 14 estuaries designated by the Commonwealth of Massachusetts that have impaired water quality. Impaired water quality just means there's a lot of inputs coming from different sources that's disturbing its ecosystem. One thing they did was install a sewer system. So in 2015, 2016, they installed a sewer system around the pond to divert wastewater around from the 1,400 homes away from the pond to reduce nutrient loads. What we have been doing is changing, or what people probably have been doing is measuring the change in groundwater nitrogen from measuring the wells. So as you see on this picture here, this is our little pond, and each white box here is a location for our wells. So we sample from the wells, and over time we're able to see how it's changing. Another thing about Little Pond is it's a tidal pond, and so what's happening is that tide is coming through these inlets. So this is the pond side, this is the sound side, and the tide's coming through this inlet for the exchange. Seagrass, algae, nectin, and detritus greater than 5 millimeters from Little Pond to Vineyard Sound. So pretty much we have our nitrogen input or inputs, mixing in Little Pond, and I'm going to be measuring the nitrogen output to Vineyard Sound. So why is this important? So for building blocks of pretty much anything, especially our estuaries, right? Carbon and nitrogen. Carbon is changing the chemistry in the estuary, making it, more impaired, changing things around. And then nitrogen is limiting the productivity in coastal marine ecosystems. So being able to understand upon ecosystem recovery is a big factor as well. Another thing about nitrogen is that with excess nitrogen, there's more respiration and more photosynthesis. So this could lead rise to the algal blooms, lower levels of dissolved oxygen in the water, and then loss of yogurt bed throughout the pond. And then you got little guys like this, like little hermit crab that get to suffer. So understanding the pond ecosystem recovery, right? So that's where the sewer construction once again comes about. So once it's installed, pretty much how long will it take to recover? This helps us understand how these damaged ecosystems respond to excess nutrient transport and being via this inlet. And overall, probably some extra storage over the years, just excess accumulation building up, that's probably caused this impairment. And then you're dealing with things like the crab stuff, right? Or you have gases in the bottom get trapped and then release, and then that's releasing more nutrients into the water or more accumulation of things. And then some recovery treatments. So pretty much like I said, the sewer system, we're trying to remove nitrogen from the water. That's gonna help us understand how these damaged ecosystems respond. And then some common misconceptions, aquatic ecosystems need more nutrients to grow. I think we all say that's false. For millions of years before our time, aquatic ecosystems have been able to be able to live and thrive and have self-sustain the whole time. And then that's dependent on humans, which is also false. They are not. Okay. I got it. All right, so now we're gonna get into our methods portion. So I sampled the inlet. So we start out with constructing my net here. Here's that McKinnon right there. He helped me construct the figures and get the foundation started. And then for the most part, I was able to take the net that we acquired and cut it out and fit it to my framing of my square structure. And then once I got that built, I was able to start sampling. So pretty much it was a 12-hour sampling day. And for every hour, I sampled for 15 minutes. So I started at 9:30 in the morning, sampled till 9:45. There's my 15 minutes. Then I waited the next hour to sample again. And I did this depending on which way the tide went. This is showing me the sound side. So as the tide's going out, collecting things that are going to the sound. And then if the tide flipped, I would just go to the other side and do the same process. And then whenever I was done sampling for the 15 minutes, pick up my net, then I collect all material, bag it, label it, and I even got to catch some cool things like this little puffer right here. Little cute guy. So before we get more into like the methods, I want to talk about this. So this is a little pond tidal record, and this is just comparing it to my sampling window just to show you. So pretty much in this graph here, we have the water level relative to the mean on the y-axis, and then the 3 days leading up, July 21st, 22nd, on the day I sampled, 23rd, and 24th. So that's just giving us time. And so you can see here on the big strike up, at high tide, a low tide, and then right around middle high tide on my day I started. So most of my data came from a falling tide, right? And you can see how this correlates to my sampling window. So right about here is about 11. Right there is about 11. And you can see as it's going down, and I'm taking my measurements before I use sampling time, it's going down. So it correlates with each other. And a little change here, it goes down again. So then we got into sorting once we figured all that out, right? So the material that we collected, we bagged up, we put it on ice, brought it back to the lab, and we categorized it, right? We sorted it out, we put it into these 10 little dishes with these labels. Oh, and I'd like to mention before we did this, we measured out the weight of these. You had to make sure we got true data. Anyway, we sorted these out in between the seagrass, algae, even have nectar and fish combined now, and the macro try sort of. And then like earlier, this is the little buffer. And then the next step in the process was dicing. So, and my buddy Akil right there, he helped me out with the dicing and grinding process. For dicing, any of the nectin and or fish or combined, that's what I had to dice. They weren't dry enough or able to be dry enough to be able to grind with the pestle mortar. So I had to dice these up with a real fine razor. It's a little tiny bit to get myself. And then for grounding was mainly the stuff that able to dry out better. So we had seagrass and algae, those worked wonderful. And then the other things are the detrital material that worked well as well. And then once I grinded up my samples, put them into these vials with labels, I took all my little vials and stuck them into a desiccator just to keep moisture out, so that way it didn't mess with my the weights that I took prior to putting it. Then the last step of what I had, or the last part of my methods, was the elemental analysis. So in the beginning portion, I had to take all them samples you've seen that were ground up in the vials and put in the desiccator. I had to take those and pack these into these little tiny tin capsules, right? I weighed them, and then I zeroed it out, put a measured amount of sample in there, measured it again, got my weight, and then that moved me on to the folding. So, I mean, I took these little tiny tweezers, tiny tweezers, and just folded up into a little tiny circle, little packing sample, right? Place those into my dish here, so that way I know how they're organized. And once I got that all situation, then put those into the carousel. Carousel was loaded up, put on top of our elemental analyzer. The sample was dropped into the chamber here, it was heated up, and then they gave me my reading on the measures of CO2. So some of the results. So what this graph is showing is the overall cumulative dry weight. So overall, I had about 140 or so grams of dry weight collected at all the material that I got on the falling tide. So, but as you can see, algae and seagrass made up most of that mass. Algae 95.34 grams, seagrass 42.51, the detrital material 2.35 grams, and then nectin also combined with fish made up 3.5 grams. And so now that you've seen what I have caught and how much of what I caught, we can talk about the more specifics here. So this is dry weight of material collected over each sample time, and this is going by the type of material. So the biggest on the y-axis we have our dry weight in grams, and on our x-axis we have the time per hour that we sampled. And you can see on each graph the values are very different. This is 0.0 grams to 0.4 grams, 0 to 15 grams, 0.0 to 0.6, and so on. But I don't want you to focus on that. What I want you to focus on, as you can see, just like I said before, on seagrass and on algae is what I most collected. And then it's 8 o'clock sampling time, that's the trend. That's when I collected most of my material throughout the whole day. So now I'm talking about how much of what each I caught. Let's talk about the elemental composition of material transported. So these graphs right here, on the y-axis we have a percent nitrogen, percent carbon. And then on x-axis, we have a material type. And so what this is showing is just the general average composition of each type of material that has. So on average, each fish had about 9% nitrogen, had about 37.1% carbon, and which makes sense. And so as you can see, the nectin in the fish, even before I combined them, I had it separated. They have a high nitrogen and a high carbon percentage. But then algae, seagrass, and other have a low nitrogen percentage and a high carbon percentage. And so now we're talking about the percentage and composition of material transported. So we have overall dry weight percentage, overall carbon percentage, overall nitrogen percentage. And once again, which is another trend here, algae and seagrass make up most of these components. So most of the dry weight came from algae, 66%, carbon 63%, nitrogen 66%, and the same for the rest. So yeah, this is the bougie slide right here. So net transport of carbon nitrogen outgoing time. So pretty much what this is showing, on the y-axis we have carbon milligrams per minute, x-axis we have time, and nitrogen same thing, nitrogen milligrams per minute, time on the x-axis. So what this is showing is that as the tide is increasing going down, we're collecting more material. Oh yeah, hold on, let me. Bam, there it is. So this is my sampling one that I was showing y'all earlier, right? So 11 o'clock, 11 o'clock. So as the tide is going down and we're picking up flow, collecting more material. And right about 1300, about 1 o'clock, things were evening out, flattening out. So that's why we have 0. And as it picks up, right around 2 o'clock, we start collecting more material. And this shows around peak flow time is around 3,600 milligrams per minute, which is, I mean, it's quite a lot. And then if you compare that to our nitrogen, almost quite similar. So 11 a.m. flow is picking up, collecting more material, down, in and out, collecting less, and then so on, going back down again, picking up. And this works. Oh, not sure. So now this next slide shows the comparison of the macro particulate material, which is greater than 5 millimeters versus the fine particulate material. So just comparison of particulates, right? So on the y-axis, we have our average concentration in milligrams per meter cubed. And on our x-axis, the so we have carbon and nitrogen. And then blue represents the large particulates, which I got. Small particulates are data that other people on the project collected. And this comparison is just showing that the stuff that I collected, the large particulates, is making up, you know, anywhere from 10 to 20% of the overall flux in the pond. And that's for carbon and same thing for nitrogen. So some conclusions and factors that may affect my data. So overall, I found out that transport of large particulate material is important. And then we also found about 12 to 13% of the carbon and nitrogen transported on the ebb tide was from macroalgae, seagrass, and detritus connected greater than 5 milliliters. Some caveats or limitations to my project. There was only one sampling day. I think more than one sampling day would give me better data. I think depending on what I collected, how much I collected may vary seasonably. Right now we're in a warmer time, but I'm like wondering how it would look when the weather starts getting colder and maybe algae production goes down. And then another caveat, the phase and strength of the tide. If y'all remember earlier on my sampling day, I forgot to mention it, but I had one of the lowest peaks of low tide on my sampling day. And I remember the day prior on the 21st, we had a full moon, so that, you know, might have played a tie or an effect into what's going on in my actions there. And some future ideas, yearly and seasonal sampling, kind of like I mentioned earlier, but just having a more consistent and current analysis over a larger span of time. Collecting more ingoing tide data. A lot of my data was on the outgoing tide, which is still fine because we got a full outgoing tide time. And then other people can use my methods for other bodies of water. Like there's 14 estuaries here in Falmouth, but there's plenty more all on the Cape. So you can take my net and stick it in anywhere, any inlet you want. Like, you want to look at it and analyze it. And so I didn't really have this prepared, but I just want to say thank you to really everybody here today. I don't think if not one of you were here, I wouldn't be here today, whether it be staff that was already here beforehand that set up, you know, the opportunity that I had, PEP students, Anji, my mentor, Emma. Y'all don't realize how much y'all really made me love science. I know that this is what I want to do the rest of my life. Even if it's not ecology or environmental biology, I won't leave this ever. And it's because of y'all. So thank you. And if there's any questions, I'd love to try to answer them. Yeah.
What was your favorite part of the process, whether it be like the making the net or like the grinding, the sampling? Like, which was your favorite part of all of it?
Honestly, it wasn't the work. It was being able to work with everybody else. Akil came in there when I needed him, and he helped me dice up. Butter came in there, helped me grind up. Emma sat there on hours and helped me develop the grass. He did the same thing. That was probably my favorite part. Just being able to grow with them, and hopefully they can see me grow as I continue my career. Not the work. The work's cool. That's all cool. The installation's cool. The data's cool. But the connection that I made, man, I just don't ever want to lose them. That's the most important thing to me. That's the coolest thing. Any other questions? Yes, ma'am.
Do you find any trash
Any trash?
in your nets?
Oh, no trash. But one thing that happened that I think was pretty funny is that Ken actually showed up to check on me about halfway through my sampling day. And we had one of those, I'm just gonna call it a water bird, pretty much dived underneath the culvert of the channel and came flying out at such a rapid speed, he could have went through my net and I could have collected a bird. Not rounding him up. I know my back next probably. Any other questions? Yes, sir.
What was the most, or I guess the most intriguing thing that you found in your net?
Most intriguing thing? Ctenophores. Those are really cool. It was unfortunate that I wasn't able to collect and preserve good samples. Because you know, over time it just kind of dissipated or just kind of went away. But that was probably the coolest thing, being able to see like, what the hell is that gelatinous figure? But then again, I know what it is, so I'm able to recognize. And then probably the puffer fish. I used to own an Egyptian puffer, so they've always been a thing in my life that I've enjoyed. That's probably the most intriguing things. Sorry, I'm in plural there. Any other fine, I love those questions, really hard, really tough. Any other questions? Yes, ma'am.
So I saw that your net was only like sampling a certain area of the
Mhm.
inlet. Is there a way that you like accounted for the rest of it? Is
Any what?
there a way that you like accounted for like the rest of the material going through that you didn't catch in your net?
Yeah, unfortunately, there was no way for me to account for it. So I mean, I could probably we have a percentage of area and the culvert that I sampled from, so we could take that and compare it to the whole width of the channel, and then we could probably get an estimated number of how much material is flowing through the full channel, not just what was coming in my net. That answer your question? Wonderful. Any other questions? No? All right, thank you.
So, hello, my name is Arlene Martinez. This summer, I was in Italy. So my summer research was to exploring the squaliform during biology diversity. So what is a denticle? So denticles are placoid scales that can be described to be like skin teeth. And the reason why we say skin teeth-like is because these denticles are all over this shark's body and are literally made of like the material of our teeth. And so some people may think that shark skin are to be smooth and silky, but unfortunately that is not the case. When we put these skin patches or when we look into these skin of the shark, this is what we see under the microscope. Now, what you're seeing are skin patches of the shark. And the circle structures that you see, those are the denticles that I'm referring to. Now, when we detach these from the skin patches together, these are the denticles that my project is focusing on this summer. And so this is a more demonstrated display of like how these denticles are, how small they are, and as well as like showing like where they are on the body. And so why is this important? What can we learn from denticle pathology? So shark bodies, since they are made with cartilage, they're not bone, it's really hard to preserve this type of skeleton. So how can we overcome this barrier? Well, denticles are to be well preserved and well mineralized to have this type of fossil records. And these denticles can be used for many things, and one of them is evolutionary history, in which one of them is diversity and evolution. So we want to know how diverse these denticles have changed over time, and as time progresses, we know that things change, right? So why not find out about our shark community as a whole? And then understanding of our shark paleontology is relating it about how our fossils, how our denticles are now to compare to how they were 100 million years ago. So we want to see how the relationship has changed, or if there's been nothing much has changed at all. And in the third bullet point, it's just to see how these denticles interact with other species as a family or as a clade, or even interacting it with other organisms as well. And so my project, I did this phylogenetic tree that was given by my mentor, and I shrunk it up to the order that I'm focusing on, which is the order Squaliforms. And this Squaliforms, it is basically a clade of deep sea sharks. And my focus was to do 6 families. And the reason for why I picked Squaliform as my choice was because since they are deep sea sharks, I wouldn't really see any type of differences or any type of like big diversity with them. So I would want to see that they're doing the same thing as fossils back then, how they were, and just to be settled as one thing only. And so that's why my job is to look at the physical morphology of these sharks. And my focus is to focus on the order Squaliforms as a whole. And so my research questions are, how does denticle morphology vary within the Squaliform order and compared to other orders? And are Squaliform denticles similar or different to fossil denticles? I also forgot this tray. So this tray is basically showing how small these denticles are, and as well as a 3D model as one of our denticles as well.
So I have two hypotheses for my color research. For one of them is the orders on the form has everything close within the clay, but different morphotypes between other clays. And my second one is the diversity of mucose of the orders on the forms all have similar relationships compared to children. So basically saying that their skin won't change or won't be diverse. And so the tentacles that I worked on was collected by my colleague Leah, and Leah partnered up with Harvard to collect skin patches. And basically what we're trying to do as like a main side goal is to build a reference catalog to be sent back to the museum as a collection for them. And the way that she did it was she picked different locations of the shrike. So for example, like letter A is the snot tip, or T is for the back fins, or M is for the material fin. She got all of these different locations of the sharks because all of the tentacles on the sharks are looking different. So we want to see how different they are from each other. And so the whole process of me going with these tentacles is three main steps. It is either imaging, bleaching, and picking. The imaging comes before bleaching and after picking. So imaging is a big, it's just a process where we can take an image of the skin patch and how they're organized on the shark skin itself. And as well as using it for future research, framing like how your scales are. Well, we are with hydrodynamic future research as well. And then for bleaching, bleaching is basically trying to detach the tentacles from the skin patch. And with that, what I did was I actually 10% bleach. Then put the skin patch onto a ether. And then this process is about 5 to 8 hours. And then whenever I see that the skin is gone, we will get an end product of tentacles only. And so when we get the tentacles by itself, we'll put it in this oven, we'll let it dry overnight. And then the next day I'll start doing the picking process. The picking process is where I would get one of those picking trays and try to pick one tentacle at a time where the tentacle came from. So each of those squares that we see on that tray basically demonstrates the different locations of where they came from that shark's body. And so how the tentacle morphology vary within this order. And so I'm going to bring back the phylogenetic. The phylogenetic is a great display to show more with our naked eye to see how these tentacles are to be so different. So what we see are like they're very so diverse. So we see different type of shapes, sizes, and even like the numbers of tentacles that are located on the skin patch. So you can see this one has many tentacles. It can probably have like thousands, and this one doesn't really have many tentacles. Are like 30, 20. So we can see that they vary so much with each other, even though they're from the same clay, they're so diverse. As you can see, there's like more circle-like, there's more of a spine-like, more like a diamond shape. And so my first hypothesis was to say that the Ortus forms has similar tentacles within the clay in different morphotypes, but my hypothesis was this was wrong. As you can see, they're to be more diverse than what I expected. So with this, what can we do with this type of information? With this information, we go with our analyses. So there are about 44 characteristics and traits that we take in with each tentacle that we see. So either they have any type of ridges, or we take any type of shape that they form. So we have 4 basic shapes. So one of them is linear, which is more of a tentacle. Geometric, we can see a dimple in the middle and a diamond shape form. Smooth, where it is only smooth texture, no ridges or anything like that. Spine is whenever they are more specked up and we see some type of. And so with this characteristic, we try to consider how the tentacles look with each other. And so when we take all of that analysis part and all that hard coding into our Excel and whatnot, first, I want to also demonstrate all the sharks that I've done all summer, and as well as a zoomed in picture of how these tentacles are looked on the shark's body. So for example, these are some of the sharks that I worked on, and these are how the tentacles look like on the body. These 2 sharks look to be more familiar because of the circle shape, the oval shape, the more spade-like shape. But to compare to these two sharks are to be seen to have more spiky-like, not to be having like a circle form. So, and as well as these two are to be seen to have not a lot of tentacles on their skin patches, but less than the other ones that we saw before. And as well as these two sharks, this one has more tentacles to compare to this tentacle. And then the last one where there's a lot of tentacles and then a close-up look at all of the 32 grids that I worked on this summer as well. So these are all the grids, and a zoomed in look. There's a more of a spine look. These are bigger than the ones that we saw before. So like for example, this is from one shark only, and you can see that there's big shapes, little small shapes, little sizes. Here one. So what do we do with this? What do we do with this kind of collection, right? So I put it into this MDS plot, and this MDS plot just helps us to see how related they are with each other with these tentacles. So on this MDS plot, I put 5 families out of 6 because of time. So basically, this MDS plot tells us how close they are to each other, right? So the overlapping and the dots being closer together is because they are similar to be with each other. And then if there's no overlapping or the dots are being more separated with each other, that means that they're to be more different. So we can see that the family Oxynota, which is the blue one, is to be so diverse with the other families, right? To compare to this family, it's more Turpida, I hope I'm doing that right, is to be more isolated. And that could be a clue too, because maybe that species isn't to be in like other landed areas. So it's to be one area only, to compare to the Oxynota, where it can be in rough areas, it can be to smooth areas, it can be like used for protection. And so that is one way to use MDS plot to see it more as a visualized look of how they So I also tried to put more data with our fossils, which my colleague Elizabeth is doing. And so as we can see, my tentacles are more modern. And when we input them with fossil tentacles, they're to be so different with each other, right? So that gives us a clue that, you know, our modern tentacles are not to be so like how they were 100 years ago. And as well as these 3 graphs as well are the same graphs, but looking at a different point of view. Since this MDS plot is a 3D space model, we can look at it at a different point of view at a 90 degree. So on graph A, we can look at it at one point of view. At graph B, we can see another, and as well as graph C. And for the reason why I say different point of view is because for graph A, we can see that, oh, like modern tentacles and fallen tentacles are to be seen to be separate from each other, but because of different traits, how their tentacles are to be looked. But then if we turn it around to a more 90 degree in that space model, we can see that in other ways, they're to be more similar to each other in some traits that they have with the tentacles together. So that gives you a clue, like you know, some traits that can be different with each other, but then in some ways it can be similar. And so my second hypothesis was the diversity of tentacles for the order Squaliforms will have similar morphotypes compared to the fossil tentacles in which we see here, right? But like that can also be like kind of debatable because here we see they're also different. So it's like an either or. And so overall with my summer research is that we are to be seeing that the Ortis polyform is to be more diverse than expected. There is to be more linear, smooth, spine, and geometric within this whole clade. And as well as that there is differences with the squaliform order tentacles to our follow tentacles, but then we can also see some similar traits that they are with each other as well. And so for my future directions with the summer research is to collect more data so that we can add not just deep sea sharks, but as well as open water sharks and follow dweller sharks to see it more to be expanded, and as well as expanding our phylogenetic tree to see it more with the visual eyes as well. And then as well as adding more of our our fossil tentacles as well to see if there's any type of changes throughout our evolution history. I acknowledgements are my mentor. She's helped me through all my my summer research this whole summer, with my brain being fried. And as well as Audrey and my PEP cohort, and as well as the people from Harvard and from Florida. This is a team tentacle that we call each other. We come together and see what we're doing and try to catch up and try to see if we're good. Any questions?
I know you said the bleaching process takes 5 to 8 hours. So would you like do that in the morning when you get to work and then like at the end get all the denticles out, or like how would you structure that?
Yeah, like I'll start bleaching like at 9 a.m. and then they'll be done around like 4 or 3 the most. There were some that just wouldn't dissolve until the next day.
So could you just leave it overnight and like the denticles would be fine?
Yeah, just like to take out like the bleaching and everything like that, and I'll just what it is.
Good job. Proud of you.
So kind of like how these tentacles form over time. I mean, it takes a lot of years for them to, I guess, like change up their morphology. But you were saying how, different sharks are in different locations, maybe different depths or more in the pelagic zone, so maybe more open oceans. Do you think when sharks specialize, let's say, going on currents more, or just kind of staying low to the floor where there isn't too much flow, do you think that has a good impact on tentacle morphology?
Yeah, like the tentacles are more smooth, but because they're kind of used to for sharks can swim faster. Compared to the tentacles that are more sharper, like the spine one, they're to be used for like protection wise. So the tentacles, like the traits and everything like that, does come into effect, like how the sharks use their skin as a whole, or even like where they're from as well.
So for the comparison for the fossil tentacles, were you using fossilized versions of the squaliforms or just general fossilized?
It was just general. From my colleague Elizabeth, she was looking at more like eons. So I think it was where like where our climate was like to the very top of like the hottest point. So I tried to input her data as well, and as well as my colleague Leah as well. So yeah, it wasn't just orders. The focus on more the orders qualified, but as well.
Talking about your MDS plot, how some, attributes were similar to fossils and some that were not. Do you know which traits were more similar versus different? Is it like more of the shape or the clustering, or is that like with your research?
Like the loss on the event, like which is which?
I was just wondering, like, if you knew like which traits are more similar between like modern denticles and fossil denticles, versus which traits are the ones that are like causing that dissimilarity.
Oh, that's kind of tricky to be honest. I think it's more as like, I think we still need to look into that.
Okay.
Because it just comes all together, but I think there should be a way to trigger what traits are similar to each other. But right now, like, just
All right. First graduate is Alex Brown. Oh, my God, yes. Next, Emmanuel Homogi. Michaela Harris. Next, Enzo Enestroza. Take a nice group photo at the end of it. Amari Johnson. The page. Let Martinez. Martin Fuller. Catlin Payne. Shaw. Akil Smith. Adeline Stewart.
No, that's all right. That's all right. We'll get you.
Sinclair Strong. Jordan Barrett. Aaron Wickware. How you guys feel?
Graduated.
Anyway, so I wanted to just say a final few words to awesome 2024 PEP cohort who I'm going to miss very dearly. And yeah, I'm probably gonna cry. You guys have been absolutely amazing this summer. Every year, you know, when the PEP students come in, it's like the highlight of the summer. You guys always do so well, but you guys have been an exceptional class. In the very beginning, you have impressed me and impressed the staff and the members of the community. And this set of presentations was just a culmination of what we knew you could do. And it's amazing to see how far you all have come from your intro presentations about yourselves and your midterm presentations when you kind of knew what you were doing and maybe not, then coming up here today and showing us that you all worked really hard this summer and did incredibly well. It is my absolute honor to have spent the summer with you. I wish I could have spent more time with you. And I hope that you all will find your way to come back to Woods Hole sometime in the future. I hope that you all will continue to be in communication with each other, continue to support each other over your careers, continue to be in touch with myself and the other PEP staff members and your mentors so that we can support you in your career. So I know all of you are going to do amazing things. You guys are absolutely phenomenal. So I want you guys to give yourself another round of applause for all of your hard work and for being so awesome. Organizations that make up PEP, the 6 institutions in Woods Hole, the Woods Hole Oceanographic Institution, NOAA's Northeast Fisheries Science Center, the Woodwell Climate Research Center, Sea Education Association, USGS, and our host for today, the Marine Biological Laboratory, in addition to the University of Maryland Eastern Shore, where you all know you're getting your academic credit. I want to say a huge thank you to all of your research mentors. So all of the PEP mentors who are here and those who are online and watching later, we absolutely, literally could not have a program without you all. So thank you all for taking the time to mentor the students, to be advisors to the students, to take them from maybe not having any idea what they're doing to giving these awesome presentations. Thank you for all of your hard work and your support of these students throughout the summer and beyond. I know many of you will be in touch with your students and continue to support them. I want to thank specifically again MBL and Rich and others for helping us host this. Thank you to the PEP staff, Dr. Gerald, who unfortunately had a family emergency and couldn't be here today. He sends his best. Dr. Harden for running an awesome class, and a huge and incredible thank you to Autumn, because Autumn made this summer work, and without Autumn, I would have been an absolute mess for sure. So thank you, huge thank you to Autumn. I want to thank all of you students again for putting in the hard work this summer, for doing your best, and also for your patience with me and my little one who likes to pop it the most. As you know, I had to drag her around with me all summer, and I appreciate you all for being just so encouraging and supportive of that. It's been a big change in my life to become a mom, and you guys have really been wonderful to me as I'm trying to navigate doing that and still being with you all. So thank you for that. Thank you all for being here today. I hope that you all will stay for lunch. It was supposed to be outside, but I think we may need to move it inside. So give me a couple minutes to get that organized, but definitely hang out, celebrate, enjoy the rest of the day. Know that you all are amazing and thank you.
We give you a group hug.
A group hug.
Yeah.
You just want it.
Yamini, I'm also behind you.
Yeah, right.